Golgi apparatus. The dynamic organelle called Golgi apparatus or Golgi complex completes post-translation modification and proteins produced in the RER and then packages and addresses these proteins to their proper destinations. The organelles was named after histologist Camillo Golgi, who discovered it in 1898. In Golgi apparatus consists of many smooth membranous sacs, some vesicles, other flattened, but all containing enzymes and proteins being processed in most cells. The small Golgi complex are located near the nucleus. As shown on figure 2 to 13, the Golgi apparatus has two distinctive feature sites or faces formed by the complex traffic of vesicles within cells. Material moves from the RER cisternae to the Golgi apparatus. A small membrane enclosed carries called the transport vesicles that are transported along the cytoskeleton polymers by motor proteins. The transport vesicles merge with the Golgi receiving region or cis phase on the opposite side of Golgi network at this shipping of transphase. Larger sacules or vacuoles accumulate, condense, and generate the vesicles that carry completed proteins produced to organelles away from the Golgi. Formation of transport vesicles, secretory vesicles, is driven by assembly of various coat proteins, including catherine, which is also regulated in vascular traffic to though and beyond the Golgi apparatus. Forward movement of these vesicles in the cyst Golgi network of saprioles is promoted by the coat protein COP2, while retrograde movements in that region involve COP1. Other membrane proteins important for detecting vesicles from include variations of RAV proteins and other synthesized enzymes. Transport vesicle and secretory vesicles and shipping region. Transport vesicle lumen of cisternae filled with secretory product. For detected vascular fusion of includes various RAB proteins and other enzymes, receptors and specific binding proteins and fusion promoting proteins that organize and shape membranes depending on the activity of these proteins. Cis proteins, vesicles and other directed towards different Golgi regions and give rise to lysosomes and the secretory vesicles for exocytosis. As indicated on figure 2 to 14, Golgi sacules at sequential locations contain different enzymes with different cyst, medial and trans levels. Enzymes of the Golgi apparatus are important for glossylation, sulfylation, phosphorylation, and limited proteolysis of proteins. Along with these activities, the Golgi apparatus initiates packaging, concentration, and storage of secretory products. Protein movements through the Golgi and control of protein processing are subject of active research. Secretory granules originating as condensing vesicles, and in the Golgi apparatus, secretory granules are formed in cells in store and product until its release by the exocytosis is signed by a metabolic, hormonal, and neural message regulated secretion. The granules are surrounded by the membrane and contain a concentrated form of secretory products. The concentration of some secretory granules may be up to 200 times more concentrated than those of cisternae of RE. The concentration of secretory granules or granule than the cisternae of rough endoplasmic reticulum. Here, exocytosis and membrane proteins enter secretory exocytosis, rough endoplasmic reticulum, and then cis Golgi, then medial Golgi, transport vesicles, and then trans Golgi, cis Golgi, and trans Golgi, lysosome, secretory granules, and exocytosis. In the rough adipose reticulum, new proteins are translocated in area cisternae. Reassembled monorich oligosaccharides are added to specific asparagine, including aniline. Proteins are folded, guided by the chaperones with strict quality control. Disulfide bonds and found between the specific cysteine residues in the Golgi network. Vesicle movement from the area and forward through the angier is promoted by the COP2. The trans-Golgi network is added amino sugar to certain oligosaccharides. Sulfur and triosine reduced to some sugar occurs. Specific vesicles with different destinations are separated and stored. The main molecule process are listed in the right, with the major compartments where they occur in the trans-Golgi network. The proteins and glycoproteins combine the specific receptors to guide them to the next stage towards their destinations. TEM, one of the area of a pancreatic acne cell shows numerous mature electron-dense secretory granules in association with condensing vacuoles in the Golgi apparatus. Such granules form the continuous of the Golgi apparatus become more condensed. The H and E stained sections secretory, the hematoxin and eosin stained sections secretory granules are often shown in intensely eosinophilic structures, which is polarized epithelial cells are connected at the region prior to exocytosis. Secretory granules then dense secretory granules with dense contents of digestive enzymes are also referred as zymogen granules. Lysosomes. Lysosomes are site of granular digestion and turnover of cellular components. Lysosomes, solution, and stoma body are membrane-limited vesicles that contain about 40 different hydrolytic enzymes and are particularly abundant in cells with great phagocyte activity. For example, macrophages, neutrophils. Although the nature and the activity of lysosome enzymes are vary depending on the cell type, the most common are acid hydrolysis such as protease, nuclease, phosphatase, phospholipase, sulfatase, and beta glucuronidase. As can be seen from the list, lysosome enzymes are capable of breaking down most micromolecules. Lysosomes, which are usually spherical, range in diameter from 0.05 to 0.5 micrometer, and present a uniformly granule electron phase appearance in the TEM electron microscopy. The macrophages and neutrophils. C. Golgi apparatus. G. Such granules of Golgi have become concentrated. C and G. Transition electron microscopy. Cystosolic components are protected from these enzymes by the membrane surrounding lysosomes, and because the enzymes have optical activity in an acidic pH, any leaked lysosome enzymes are particularly activated in pH of systole 7.2 and harmless to the cell. Lysosome hydrolyses are synthesized and segregated in the RER and then transferred to Golgi apparatus, where the enzymes are further modified and packaged in vacuoles that form lysosomes. The marker monosphix phosphate MA6P is added by a phosphotriferase in the cis Golgi only in the N-linked oligosaccharides of the hydrolase destined for lysosomes. Membrane receptor for M6P containing proteins of trans-Golgi network then binds these proteins and divert them from the secretory pathways for segregation into lysosomes. Material taken from outside the cell by endocytosis is digested when the membrane of the phagosome or pinocytic vesicles fuse with the lysosome. The mix of endocytosis material with the lysosome's enzyme and activates proton pumps in the lysosomal membrane that acidifies the content, allowing digestion. The composite active granule is now termed as secondary or heterolysosome. Heterolysosomes are generally somewhat large and have a more heterogeneous appearance in the transmission electron microscopy because of the wide variety of materials they may be digesting. Lysosomes are spherical membrane enclosed vesicles that fuse as sites of intracellular digestion and are particularly numerous in cells activated after various types of endocytosis. Cells of the kidney show numerous purple lysosomes in the cytoplasmic area between the basally located nucleoli and apical ends of cells. Nucleoli of developed using endocytosis. These cells actively cake up a small protein subluminous developing to lysosomes and the release resulting amino acid reuse. During digestion of macromolecules, released nutrients diffuse into the cytoplasm through the lysosomal membrane. Indigestible materials is retained within a small vacuole remain called residual body in some long-lived cells. For example, neurons, heart muscle, residual bodies can accumulate over the times as granules of lipofuscin. Besides the degradation exogenous macromolecules, lysosomes also function in a process called autophagy, by which excess autophagosomes, which then fuses with the lysosome for digestion of the enclosed material. Autophagy is enhanced in times of starvation or nutrient stress. Amino acids, nucleotides, and other digestion products are released from autophagosomes for reuse and source of energy in a new synthetic reactions. Autophagy is a process in which the cell uses lysosomes to dispose of excess non-functional organelles or membranes that emerge to the SRR enclose the organelles to be destroyed. Cell process in which useless lysosomes dispose of excess non-functional organelles membranes. In this TEM transmission electron microscopy, the two autophagosomes in the upper left contain protons of RER more electron dense than the neighboring normal RER in the center. Contained mitochondrial membranes of NER is shown on vesicle. Organelles are large aggregates of non-functional micromolecules in cytoplasm are degraded. Figures 2 to 117 and 2 to 18. During autophagy, a lipid bilayer membrane forms around and isolates the cytoplasmic portion to be removed, producing an autophagosome, which then fuses with the lysosome for digestion of an enclosed material. Autophagy is enhanced in times for starvation or products are released from the autophagosomes to reuse as source of energy in new synthetic reactions. Medical application. Disease categorized as lysosomal stage disorders stem from the defect in one or more digestive enzymes present in lysosomes, usually due to a mutation leading to a deficiency of some enzymes, or defects due to faculty post-stronal processing in cells that must digest the substrate of the missing of defective enzymes following autophagy. The lysosomes cannot function properly. Such cells accumulate large secondary lysosomes or residual bodies filled with the indigestible macromolecule. The accumulation of these vesicles may inadvertently interfere with the normal cell tissue function, producing symptoms of the disease. A few lysosomal storage diseases listed in Table 2 to 3 with the enzymes involved in each and other tissue affected. Proteosomes are abundant. Cytoplasmic and nuclear protein complex are not associated with membrane. Each approximately is the size of a small orthogonal subunit. The function to degrade, denature, and otherwise non-functional polypeptides. Polypeptides also remove proteins no longer needed by the cell and provide an important mechanism for restricting the activity and provide an important mechanism for specific protein in a certain window of time. Whereas lysosome digestion organelles are membrane by autophagy, proteosomes deal primarily with free portions as individual molecules. As shown diagrammatically in figure 229, the proteasome is a cylindrical structure made of four sacked rings, each composed of 7 proteins including proteases at each end of the cylinder in a regulatory protocol that contains ATPase and recognized proteins with attached molecules of ubiquitin in abundant cytosolic 76 amino acid proteins found in the cells, misfold and denatured proteins of short-lived proteins with oxidized amino acids are recognized by the chapters and targeted for destruction by other enzymes complex that conjugate ubiquitin to lysosome reduces of the protein by formation of polybiquitin chain. Ubiquitin proteins are recognized by the granulatory particles of proteasomes unfolded by ATPase using energy from ATP and then translocated into the core of the cylindrical structure and degraded by endopeptides. The ubiquitin molecules are released for reuse and peptide produced may be broken down further into amino acids or they may be other specialized destinations such as the antigen presenting complex of cell activating and immune response. Medical application failure of proteasomes or other aspects of a cell protein quality control can allow large aggregates of proteins to accumulate in affected cells. Such aggregates may absorb other macromolecules to them and damage or kill cells. Aggregates released from the dead bodies can accumulate in the endoplasmic reticulum (ECM) of the tissue and in brain. This can interfere directly with the cell function and lead to neurodegeneration. Alzheimer's disease and Huntington's disease are two neurologic disorders caused by initially by such protein aggregates. Mitochondria, mitose, thread, and chondria granule are membrane enclosed organelles with arrays of enzymes specialized for aerobic respiration and produced of adenosine triphosphate with high energy phosphate bonds which supply energy for most cellular activities. Glycolysis converts glucose anaerobically and pyruvate of the cytoplasm, releasing some energy. The rest of the energy is captured when pyruvate is imported into mitochondria and oxidized into CO2 and H2O. Mitochondria enzymes yield 15 times more ATP than is produced by the glycoside alone. Some of the energy released in the mitochondria is not stored in ATP but is dissipated in the heat that maintains body temperature. Mitochondria are usually elongated structures with diameters of 0.51 micrometer, lengths up to 10 times greater. They are highly plastic, rapidly changing shape, fusing with one another and dividing, and are moved through the cytoplasm along microtubules. The number of mitochondria is related to the cardiac muscle. Cells of some kidney tubules have abundant mitochondria, where cells with a low energy metabolism and few mitochondria. Similarly, mitochondria of different share cells are concentrated in cytoplasmic regions where energy utilization is more intense. Medical application: myoclonic epilepsy with ragged red fibers is a rare disease occurring in individuals in whom cells of specific tissue, notably regions of skeletal muscle, inherit micro mitochondrial DNA with a mutate gene for lysine tRNA, leading to defective synthesis of respiratory chain proteins that can produce a structural abnormality in muscle fibers and other cells. Mitochondria are often sufficiently large to be visible with the light microscope as numerous discrete organelles under the TEM. Each mitochondrion is said to have two separate, very different membranes that together create two components, the innermost matrix or the narrow intermediate base. Both mitochondrial membranes contain a higher density of protein molecules than other membranes in the cell and have reduced fluidity. The outer membrane is a stave-like containing many transmembrable proteins called porins that form channels through which small molecules such as pyruvate and other metabolic readily pass from the cytoplasm to the intermembrane space. The inner membrane has long folds called cristae, which project into the matrix and greatly increase the membrane surface area. The number of cristae in mitochondria also corresponds to the energy need of the cell. The lipid bilayer of inner membrane contains usual phospholipids and is highly impermeable to ions. Integral proteins include various transport proteins that make the inner membrane selectively permeable to the small molecules required by the enzymes in the matrix. Mitochondrial matrix enzymes include those that oxidize pyruvate and fatty acids from acetylcoenzyme, those of the citric and cyclic of the oxidized acetyl CoA, releasing CO2 as waste and small energy rich molecules that provide electrons for transport among the electron transport chain. Enzymes that other components of the chain are embedded in the inner and allow oxidative phosphorylation, which produces the most ATP in animal cells. Formation of ATP of oxidative phosphorylation enzymes occurs by chemiosmotic process. Membrane proteins guide the small electron carrier molecules through closely packed enzyme complex so that the electron moves sequentially along the chain. Electron transport is coupled with orient with oriented protein uptakes and releases, with protons accumulating in the intermembrane space, the producing an electrochemical gradient across the inner membrane. Membrane-associated proteins of the ATP synthesis system from large 10 nanometers multi-subunit globular express on stalk-like structures that project from the matrix site from the inner membrane. This enzyme complex containing a channel through the proteins flow down the electrochemical gradients, crossing the membrane back to the matrix. Passage of protons through the channels cause rotation of a specific polypeptide in the globular ATP synthesis complex, converting the energy of proton flow into the mechanical energy of protein movement. Mechanical energy is stored in the new phosphate bond of ATP by the subunit polypeptides binding adenosine diphosphate and inorganic phosphate. A steady torrent of proteins along the gradient allows such of this remarkable synthesis complexes to produce more than 100 molecules of ATP per second. Another role of mitochondria occurs at the time of cell stress when the protein cytochrome C is released from the inner membrane's electron transport chain. In cytoplasm, this protein activates sets of proteases that degrade all cellular components in an regulated process called apoptosis, which results in the rapid cell death. New mitochondria originated by the growth and division fusion of pre-existing mitochondria during each daughter cell receives approximately half of the mitochondrial in the parietal cell. New mitochondria originated by growth division of pre-existing mitochondria. Unlike most organisms, mitochondria are partially autonomous of nuclear genes and activities. The mitochondrial matrix contains a small circular chromosome of DNA, ribosomes, mRNA, and tRNA. All the similarities to the corresponding bacterial components. Protein synthesis occurs in mitochondria because most of reduced amount of mitochondria DNA. Only a small subset of mitochondrial proteins is produced locally. Most are encoded by nuclear DNA synthesized on free polyribosome and cytosol. These proteins have short terminal amino acid sequences that serve as signals for their uptake across the mitochondrial membrane. The observation of the mitochondria have certain bacterial characteristics led to the later work to the understanding that mitochondria evolved into an ancestral aerobic prokaryotic lived. The observation of mitochondria have certain bacterial characteristics that led to the work of the understanding that mitochondria evolved from an ancestral aerobic prokaryotic that lived symbiotically within an ancestral eukaryotic host cell. Prokaryotic and eukaryotic host cell. Paroxysomes are spherical organelles enclosed by a single membrane or named for their enzyme reducing and degrading hydrogen peroxide H2O2. Figure oxidize located here. Oxidize substance by removing hydrogen atoms and are transferred into molecular energy, producing H2O2. Peroxidase such as catalase immediately break down H2O2 with its potentially damaging to the cell. These enzymes also inactivate various potentially toxic molecules, including some prescription drugs, particularly in the large and abundant peroxisomes of the liver and kidney cells. Other diverse enzymes in peroxisomes complement certain functions of the SCR and mitochondria in the metabolism of lipids and other molecules. Thus, the oxidation of long-chain fatty acids, H carbon and longer, is preferentially accomplished by peroxisomal enzymes that differ from their mitochondrial counterparts. Certain reactions leading to formation of bile acids and cholesterol also occur in peroxisomes. Peroxisomes form in two ways: binding of precursor vesicles from the ER of growth divisions and persisting peroxisomes. The two mitochondrial membranes of the innermost matrix can be seen in TEM diagram. The outer membrane is a cyste that increases the surface area gently, and the matrix is a gel with high concentration enzyme ATP synthesis. Peroxisome from two ways: budding of the precursor vesicles of the ER and growth with pre-existing peroxisomes. Peroxisomal proteins are synthesized on free polyriboxes and have targeted sequences on amino acids at either terminus recognized by receptors located in the peroxisome membrane for import into the organelle. Peroxisomes are small spherical membranous organelles containing enzymes that use O2 to remove hydrogen atoms from fatty acids. in a reaction that produces hydrogen peroxide that must be broken down to water or O2 by another enzyme catalyst. By Tm, peroxism generally shown on a matrix of moderate electrodensity aggregated electron-rich particles represent glycogen. Peroxisms in most species are characterized by a central, more electron-dense crystalloid aggregation of consistent enzymes as shown here. The small size, large number, and widespread cellular distributions of peroxisms is shown on cultured osteosarcoma cells produced for immunocytochemistry with a fluorescent antibody against the protein specific to this organelle. Cell organelles are stained bright blue in ADAPI. Medical applications. Several rare disorders arise from defective proximal proteins. Neonatal adrenoleukodystrophy is caused by a defective integral membrane protein needed for transport of very long chain fatty acids into the peroxisome for B-oxidation. Accumulation of this fatty acid in the body fluids can disrupt the myelin sheaths in nerve tissue, cause severe neurologic symptoms, deficiency and peroxisome enzymes, cause Zellweger syndrome that affects the structure and functions of several organ systems. The cytoskeleton. The cytoplasmic cytoskeleton is a complex array of microtubules, microfilaments, also called actin filaments, and intermediate filaments. This protein polymer determine the shape of the cell, play an important role in the movement of organelles and cytoplasmic vesicles, and also allow the movement of entire cells. Important properties, functions, and locations of the cytoskeleton components are summarized in Table 2.4. Microtubules. Within the cytoplasm, all eukaryotic cells are fine tubular structures known as microtubules. Most of these are highly dynamic in length. Microtubules are also organized into larger and more stable arrays called axonims. So microtubules are organized larger, more stable arrays are called axonims. Microtubules MT and actin filament MF can both be clearly distinguished in the TEM fiber cytoplasm, which provides a good comparison between relative diameters of these cytoplasmic complexes. Arrays of microtubule filaments and microtubules are usually demonstrated by immunocytochemistry using antibodies against their subunit proteins as in the cultured cell. Actin filaments are most concentrated at the cell periphery, coming prominent circumference, bundles of which fine filaments project into the cellular extension and push against the cell membrane. Actin filaments from dynamic networks form important cellular shape changes such that during cell division, locomotion, and formation of cellular promotions. Actin filaments from a dynamic network important for cell shape changes such as those during cell division, locomotion, and formation of cellular processes. Folds and pseudopodia. Pseudomicrotubules are oriented in arrays that generally extend from the centrosome and near the nucleosome, the most peripheral extension. Besides serving to stabilize vesicles and organelles into the cell periphery and dynein-based transport towards the cell nucleus. Here, properties of cytoskeleton components, microtubules and filamentous and intermediate filaments. Microtubules, microfilaments, intermediate microtubules. Alpha and beta protofilament. Diameter 25 nanometers. and maintains cell shape and polarity, provides tracks for organelle and chromosome movements, moves cilia and flagella. G action monitors two intertwined filaments of F action, 5 and 7 nanometer globular G action. Contract the movement cells, change cell shape, cytokinesis and cytoplasmic transfer streaming. Contract and move cells. So the microfilament contract and move cell polarity microtubules and intermediate filaments follow straighten cell and tissue structure. In cytoplasmic extension cell cilia discussed in chapter 4 and flagella extensions called cytoplasmic extension called cilia and flagella. Each microtubule is hollow with and diameter of 25 nanometer and a wall of 5 nanometer thick, a structure that confers significant rigidity to help maintain cell shape. Microtubules vary in length but can become many micrometers long. Two or more microtubules are often linked side by side by protein arms or bridges, which particularly important in axonemes in cilia and flagella. Axonemes flagella as indicated in table 2 to 4. The protein subunit of microtubules is a heterodimer of alpha and beta tubulin. The protein subunits of microtubule is a heterodimer of alpha and beta tubulin, each with a molecular mass of about 50 kDa kilodalton. Under appropriate conditions, the tubulin heterodimers polymerize into form microtubules, which have a slightly spiral organization. The tubule subunits are linked lengthwise as protofilament with 13 parallel profilaments forming the circumference of each tubular wall. Polymerization of tubulin is directed by microtubule organelles center MTOCs, which contain short assemblies of tubulin that act as nuclear sites for further polymerization. Microtubules are polarized structures with groove polymerization occurring more rapidly at the end. Microtubules show dynamic instability with the continuous cycle of polymerization and depolymerization at the steady state condition, which depend on the concentration of tubulin and presence of various microtubule associated proteins. Enzymes for assembly is derivated from GTP bound and incoming tubulin subunits. Individual microtubules shorten as depolarized exceeds growth. Microtubules stability varies greatly with tubular location and function. Microtubules of cilia are very visible, while those of the microtic spindle are short-lived. The dominant MTOC is most cells in enterosomes, which is organized around two cylindrical centrioles, each about 0.2 micrometers in diameter, about 0.3-0.5 micrometers in length. Each centriole is composed of 9 highly organized microtubules with their long axes at the right angles. The paired centrioles organize nearby tubulin complexes and other proteins as a pericentriolar matrix found close to the nucleus of non-individual cells. Before cell division, more specifically during the period of DNA replication, each cytoplasm duplicates itself so that now each centrosome has two pairs of centrioles. During mitosis, the centriole divides into halves, which moves opposite poles of the cell and becomes organizing centers for microtubules and mitotic spindles. Microtubules also form part of the system of intracellular transport of membranous vesicles, macromolecular complexes, and organelles. Well-studied examples include exoplasmic transport in neurons, myelin transport in pigment cells, chromosome movement by mitotic spindle, and vesicle movement among different cell components. On each of these examples, movement is suspended in microtubules is disrupted. Transport among microtubules is under the control of proteins called motor proteins, which uses ATP in moving in large structure. Kinesins carry material away from MTOC near the nucleus towards the plus and N microtubules. Anterior transport. Cytoplasmic dyneins carry material along microtubules in the opposite direction. Retrograde transport generally towards the nucleus. Important roles of system include extending ear from of plasmic membrane vesicles through the GR apparatus. Medical application. Several inhibition compounds used by cell organists to study details of microtubule dynamics are also widely used in cancer chemotherapy to block activity of the mitotic spindle in readily growing nucleoplastic cells. Such drugs include vinblastine, vincristine, and pancreatex, all of which are generally discovered as plant derivatives. Inhibitor biology study by microbiologists widely chemotherapy to block activity of mitotic spindle. Such drugs include microfilaments are composed of actin and actin subunits and allow motility and most contractile movement in cells using reversible assembly of actin filaments and interaction between these filaments associated with myosin family proteins. Actin filaments are thin, polarized polymers, shorter and more flexible than microtubules. They are composed of globular G-actin monomers that assemble in the presence of K plus and Mg plus into double-stranded helix of filamentous F-actin, G-actin, generally added to pre-existing filaments, but new filaments can be formed from a pool of G-actin by the action of nucleating proteins such as formalin. Another nucleatic factor, a complex of polypeptides called ARP23, also binds to the site of pre-existing actin filamentous and induce a new F-actin branch, a process that can lead to formation of actin filament network. Like microtubules, actin filaments are highly dynamic. Monomers are added rapidly and band end with ATP hydrolyzed at each addition. At the same time, monomers dissociate at the minus pointed end. This leads to migration of subunits through the polymer, which occurs rapidly in process called treadmilling. In cells, both the assembly and diassembly of subunits from F-action poly promoted by other proteins such as profilin and cofilin respectively. Actin is very abundant in all cells and is connected in network of actin filaments and are free G-action subunits near the cell membrane. The cytoplasmic region, often called the cell cortex, activity produced in an important branched network of microfilaments in the region. Microfilaments rich cellular extensions include surface force and ruffles are important for endocytosis. The microfilaments of the cortex underline the endosomal trafficking. Cell movements on farmed subunits substrates involve sheet-like protrusions or lamellopodia in which the contracted actin filaments are continuous with deeper parallel F bundle called stress fiber. Actin binding proteins such as foramin and other junction mentioned change the dynamic physical properties of microfilaments, particularly their length and interactions with other structures, and this determines the viscosity of other mechanical properties in local cytoplasm. Cross-linking within networks of actin, increase cytoplasmic viscosity, while serving and capping the filaments tends to decrease viscosity. The length and other physical properties of actin filaments are controlled by various types of actin binding proteins, including those indicated the figure 2 to 2.27. Just as the molecular motor, kinase, and DNase move structures along the microtubules, various myosin motors use ATP to transport cargo along actin. Movement is usually forward and based on active filaments. Myosin VI is the only known myosin that moves in the other direction. Interactions between a filament and myosin form basis of various cell movements. Transport of organelles, vesicles, and granules in the process of cytoplasmic streaming. Contractile rings of microfilaments with myosin 2 contracting the porosities to cells by cytokinesis during mitosis. Membrane-associated molecules of myosin I whose movement along microfilaments produce the cell surface changes during endocytosis. Stabilized arrays of actin filaments integrate with arrays of thicker 16 nanometer myosin filaments, permit very forceful contractions in specialized cells of those muscles. Intermediate filaments. The third class of cytoskeleton components includes filaments intermediate size between the other two, with a diameter in averaging of 10 nanometers. Unlike microtubules and actin filaments, the intermediate filaments are stable, confer increased mechanical stability of the cell structure, and are made up of different protein subunits in different cell types. More than a dozen proteins ranging from size 40 to 230 kilodalton serve as subunits of various intermediate filaments and can be localized by immunohistochemistry in various cells, as indicated in table 2 to 4. All the subunits are coiled rod-like diamonds that form antiparallel transmers, which self-assemble into large cable-like bundles of protofibers stabilized by further lateral interactions. Table 2 to 5 lists 6 classes of intermediate filament proteins forming rod-like subunits, their size and cell distributions. Important intermediate filament proteins include the following: keratins and vimentins, neurofilament and lamins. Intermediate filament proteins forming rod-like subunits, their sizes and cell distribution. Important intermediate filaments proteins include the following: keratins or cytokeratins, a diverse family of acid and basic isoformations that compose heterodyne subunits of intermediate filaments in all apical cells. They are encoded by over 30 related genes and produce filaments with different chemical and immunoglobulic properties. Intermediate filaments of keratin form large bundles tonofibers that attach to certain junctions between epithelial cells, the skin epidermal cells, cytokeratins, and accumulation during differentiation in the process of keratinization, producing an outer layer of non-living cells. Reduced dehydration. Keratinization of the skin made terrestrial life possible in course of evolution. Terrestrial life possible in course of evolution. Keratinization also provides some protection from minor abrasions and products of various protective structures, skin and nails, such as nails, as well as feathers, break horns, and vesicle properties. Vimentin is also common in class 3. Intermediate filaments protein are also found in most cells derived from embryonic mesenchyma. Important vimentin-like proteins include desmin, found in almost all muscle cells, glial fiber acid proteins found in especially in astrocytes, supporting cell of central nervous system. Desmin fibers of a cluster cell are shown in immunocytochemically in figure 1.12a. Neurofilament protein of the three distinct size makes heterodimer that forms the subunit of major intermediate filaments of neurons. Lamins are family of 7 isoforms present in the cell nuclei, where they are a structural framework called the nuclear lamina just inside the nuclear envelope. Intermediate filaments display an average diameter and weight between the actin filaments and microtubules. Between the actin filaments and microtubules, and serve medical application. The presence of a specific type of intermediate filament in tumors can often reveal cellular origin of the tumor. The presence of a specific type of intermediate filament in tumors can often reveal the cellular origin of the tumor. Information important for diagnosis and treatment of cancer. The identification of intermediate filament proteins by means of immunocytoplasmic method is a routine procedure. One example of it is used in GFAP to identify astrocytomas, the most common type of brain tumor. Inclusion. Cytoplasmic inclusions contain accumulated metabolic or other substances, but unlike organelles have little or no metabolic activity themselves. Most inclusions are transitory structures not enclosed by membrane. Features of important and commonly seen inclusions are shown in figure 2.2.28 and include the following: lipid droplets, accumulation of lipid filling adipose fat cells and present in various other cells, glycogen granules, aggregates of the carbohydrate polymer in which glucose is stored, visible as irregular clumps of periodic and acid scape, positive or electron dense material in several cell types, notably liver cells, pigmented deposit of naturally colored materials, also melanin. Inclusions also include lipid droplets, glycogen granules, and melanin. Skin preserve to prevent cells from ultraviolet radiation, lipofuscin, pale brown granule found in cells, especially in stable non-dividing cells. Neurons, cardiac muscles. Neurons and cardiac muscles. Complex of matrix of material partially derived from reduced bodies after lysosomization and hemosiderin, a dense brown aggregation of determined ferritin proteins with many atoms of bound iron, protein in phagocytic cell in the liver and the spleen, where it results in phagocytic and red blood cell phagocytosis of red blood cell. Medical application. A condition termed hemosiderosis, in which the iron in containing insulin and homostatin occurs in cells of organs throughout the body, may be seen with increased uptake of dietary iron, impaired iron utilization, or with excessive lysis of red blood cells. Hemosiderosis itself does not damage cell or organ function. However, extreme accumulation of iron in cellular hemosiderin can lead to disorders such as hemochromatosis and iron overload syndrome. in which tissue of the liver and other organs are damaged. A summary of the major structure and functional features of cytoplasmic materials in Table 2.6. So, plasma membrane cilia, short, numerous in number. Short and numerous in number. Flagella, long, singular membrane extending from sperm cells. Microvilli, numerous thin membrane folds. Nucleus, large structures. Nuclear envelope, double membrane boundary. Nuclear pores opening through the nuclear envelope. Nucleus, large. Cytoplasm, contents of cell between the plasma. Cytoplasm, between the plasma. Cytosol, viscous fluid medium with dissolved solutes. Rough endoplasmic reticulum, extensive interconnected membrane. Smooth endoplasmic reticulum, extensive interconnected membrane network lacking ribosomes. Golgi apparatus, vesicles, lysosomes, peroxisomes, mitochondria, ribosomes. Cytoskeleton, microfilaments, intermediate filaments, microtubules. Centrosomes, proteosomes, and inclusions. Chapter 3, Nucleus. The nucleus. Summary of key points. Nucleus envelope. Cytoplasm is separated from nuclear plasm by nuclear envelope, a double set of membrane with a narrow perinuclear space. The outer membrane binds with the ribosome and is continuous with RER. The nuclear envelope is penetrated by nuclear pore complex, large assemblies of nuclear pores with eightfold summary through which proteins and protein RNA complexes move in both directions. The nuclear envelope is supported internally by a meshwork, the nuclear lamella composed of intermediate filament subunits called lamins. Chromatin. Chromatin is the combination of DNA and its associated proteins. Chromatin and DNA that is active in transcription stains lightly and called euchromatin. Inactive euchromatin stains more darkly in heterochromatin. Euchromatin. The DNA molecule initially works around the complex of basic protein and called histone from nucleosomes, producing a structure resembling base of a string. Additional levels of chromatin fibers condensation are less well understood and involve non-histone proteins including saffrol or large protein complexes. The extra X chromosome in cell of female mammals form a facultative heterochromatin and can be seen on bird body. Nucleus. The nucleus is very basophilic or electron-based area of chromatin localized where RNA transcription and ribosome subunits assemble occur. By TEM, an inactive molecule is seen to have fibrous and irregular parts where rRNA formed and abnormal subunits are assembled respectively. The cell cycle. The cell cycle is the sequence of events that control cell growth and division. In G phase, the longest part of the cell brings immediately after mitosis and includes all preparations for DNA replication. The period of DNA and histone cytosis is a S phase. In a short G phase, the cell prepares for division during mitosis. Cell cycling is controlled by a sequential appearance of key cytoplasmic proteins and cyclins which bind with CDK. CDK phosphorylate and activate the enzymes and transcription factors whose function characterize each phase of the cell cycle. Progress through the cell cycle stage is monitored and checkpoints, including the G restriction point, only when each phase activities are completed are the cycling chains to trigger those of the next phase. Mitosis. The stage of mitosis cell division include prophase when chromosomes condense, the nuclear envelope deassembles, and the microtubular spindle forms. Metaphase when chromosomes are aligned anaphase, when the begin of separate towards the two chromosomes of telophase. When stage of mitotic cell divisions include prophase, when chromosomes condense, the nuclear envelope disassembles, and the microtubular spindle forms. Microtubular spindle. Metaphase, when chromosomes are aligned. Anaphase, when they begin to separate towards the two centrosomes. Telophase, when nuclear envelope reforms into separate chromosomes. Telophase ends with cytokinesis or cell cleavage into two daughter cells by a contractile ring of actin filaments and myosin. Telophase ends with cytokinesis with two daughter cells of actin filaments and myosin. Stem cells and tissue renewal. Stem cells occur in tissue with rapid cell turnover. They divide slowly in a symmetric manner. Cell tissue with a daughter cell remaining as stem cell and bone marrow, and one becoming committed towards the differentiation. Cell committed to differentiate, transit, amplifying, or progenitor cells typically divide more rapidly than stem cells before showing or stopping division and differentiate. Meiosis. Meiosis is the process by which two successive cell divisions produce cells are called gametes, containing half the number of chromosomes and found in somatic cells. Prophase of the first meiotic division is a unique extended period in which homologous chromosomes pair and undergo genetic recombination. During the process called synapsis, synaptic pairs separate towards the two daughter cells. First meiotic division. The second meiotic division occurs with no intervening S phase and separates the sister chromatids into two final cells are haploid. Meiosis. Apoptosis. Apoptosis is the process by which redundant of defective cells are readily eliminated in a manner that does not provoke a local inflammatory reaction of the tissue. Apoptosis involves a cascade of events controlled by the BCL2 family of proteins, regulating the release of death-promoting factors from mitochondria. Cytochrome C from mitochondria activates cytoplasmic processes called caspases, which degrade proteins of the cytoskeleton and cell membrane. Endonuclease are activated, which degrades all nuclear DNA. Cell and nuclear volume shrinks rapidly, and the cell membrane changes produce extensively bleeding of the cell surface. Late in apoptosis, the cell breaks into many small apoptotic bodies in the undergo phagocytosis by neighboring cells. Apoptosis occurs rapidly with little or no release of proteins that would trigger inflammation, unlike the death of injured cell by necrosis that typically induce local inflammation. The nucleus. Components of the nucleus. Nuclear envelope chromatin. Then there is medical application and medical application. Then nucleus and the cell cycle. Medical application and mitosis. Then stem cell and tissue renewal. Meiosis. Apoptosis. Meiosis. Meiosis is a specialized process involving two unique and closely associated cell divisions that occur only in the cell that it will form sperm and egg cells. Differentiation of these germ cells or gametes is discussed fully in chapter 21 and 22, but the chromosomal aspect of meiosis are described here for comparison of the events of meiosis. Two key features characterize meiosis. Early in the process, the homologous chromosomes of each pair, mother and father, come together and activate termed synapses. During synapses, double chain breaks and repair occurs in the DNA, some of which results in local DNA exchange called crossovers between the aligned homologous chromosomes. This procedure produces new combinations of genes crossover in genes new chromosomes. The cell producer are haploid, having just one chromosome for each pair present in the body's somatic cell. The union of haploid eggs in sperm at fertilization forms a new diploid cell. It can develop into a new individual. As shown in figure 3 to 18, the important events of meiosis unfolds as follows. A cell approaching the first mitotic divisions has just completed a typical S phase and replicated its DNA. Each chromosome contains the two individual DNA molecules called sister chromatid bound by cohesin proteins. During a great elongated prophase of first mitotic divisions, the partially condensed chromatin of homologous chromosomes bring to come together and physically associate along their length during synapsis. Because each of the paired chromosomes has two chromatids, genetics refer to synaptic chromosomes as tractors to emphasize that four copies of each genetic sequence are present. During synapsis, combination or crossover occurs among the four chromosomes. The mixture of the genes inherited from each parent yield new different sets of genes to be passed in the next generation. In human, spermatogenesis process normally lasts for 3 weeks. Oogenesis arrests in the mitotic phase from the time of the formation of the fetal ovary through the woman's reproductive maturity, that is about 12 years to nearly 5 decades. So 5 decades. When synapsis and crossing over completed, the chromosome become fully condensed and undergo metaphase, anaphase, and telophase, and cell divides. The first mitotic division separates the homologous chromosome with the pair during synapsis. Each of these separation chromosomes still contains two chromatin held together by cohesins at their centromeric DNA. Each of the two new cells now divide again and more rapidly with an inverting S shape. In the second mitotic division, cohesin proteins are lost and chromatids separate into opposite poles as individual chromosomes. In each new cell, nuclear envelope forms around the new haploid cell of chromosomes. In summary, meiosis and mitosis share many aspects of chromatin condensation and separation, but different in key ways. Mitosis is a cell division that produces two lipid dipolar cells. Meiosis involves the cells 4 haploid cells. During mitotic crossover, new combinations of genes are produced, and every haploid cell is genetically unique, lacking synapsis and opportunity of DNA recombination. Mitosis two cells are the same genetically. Medical application. In humans, chromosome is 21. It's a very small chromosome and some most likely overlooked. In human, chromosome 21 is very small chromosome and sometimes overlooked at the metaphase and anaphase checkpoint. Failure to those homologous chromosomes to separate non-disjunction at the first metabolic division also occurs with greater frequency in older oocytes or sperm progenitor cells. A gamete retaining this chromosome pair is visible zygote after fertilization, but the developing trisomy 21 individual has morphological and connective infirmities associated with Down syndrome. In humans, chromosome 21 is small chromosome, some and most likely to be overlooked at the metaphase anaphase checkpoint. It's a small chromosome and overlooked at checkpoint. Failure of those homologous chromosomes to separate Non-disjunction at the first meiotic division also occurs in greater frequency in older oocytes or sperm progenitor cells. A gamete retaining this chromosome, this pair, the metaphase anaphase checkpoint, is very small. The homologous chromosome failure of those homologous chromosomes to separate. A gamete retaining this chromosome pair of is variable zygote after fertilization, but the development of trisomy 21 individual has morphologic and congenital impairments associated with Down syndrome. Epithelial tissue. Characteristic features of epithelial cells: basement membranes, intercellular adhesion, and other junction. Specialization of apical cell surface: microvilli, sterocilia, and cilia. Types of epithelia: covering the lining epithelia, secretion epithelia glands. Transport across epithelia, renewal of epithelial cells. Summary of key points. Assess your knowledge. Despite the complexity, the organs of the human body are composed of only four basic types. Four basic tissue types: epithelial, connective, muscular, and neural tissues. Epithelial tissues, connective tissues, muscular tissues, and nervous tissues. Each epithelial is an assemblage of similarly specialized cell units in performing a specific function. The basic tissue, each containing extracellular matrix ECM as well as cells associated with one another in the variable proportions and morphologies. Characteristics of each organ. The main features of the basic tissue types are summarized in Table 4.1. Connective tissue is characterized by cell producing very abundant ECM. Muscle tissue is composed of elongated cells specialized for contraction and movements, and nervous tissue is composed of cell with long, fine process specialized to receive, generate, and transmit nerve impulses. Most organs can be divided into parenchyma, which is composed of the cell responsible for organs' specialized functions, and the stroma, the cells of which have supporting role in the organ. Except in the brain and the spinal cord, the stroma is always connective tissue. Connective tissue is characterized by cell producing very abundant ECM. Extracellular matrix. Cellular matrix. Muscle tissue is composed of elongated cells specialized for contraction and movements. Nerve tissues is composed of cells with long, fine processes specialized to receive, generate, and transmit nerve impulses. Most organs can be divided into the parenchyma, which is composed of cells responsible for the organ's specialized functions, and the stroma, the cell of which have the supporting role in the organ. Parenchyma, supporting role, and a specialized function. Epithelial tissue are composed of closely aggregated polyhedral cells adhering strongly to one another and to thin layer of ECM forming cellular sheath that lines the cavities of organs and covering the body surface. Forming cellular sheaths that line the cavities of organs and cover the body surface. Epithelial. Upon and thelial. Nipple. Line all external and internal surface of the body, and all substances that enter and leave the organ must cross this type of tissue. The apical functions of epithelial. The principal functions of epithelial tissue include the following: covering, lining, and protecting surface. Absorption. and secretion. Epidermis, intestinal lining, and parenchyma cells of glands. Secretion. Parenchyma glands of cells, intestinal line, and epidermis. Surface specific cells of certain epithelial may be contractile myoepithelial cells or specialized sensory cells, such as those of taste buds or the olfactory nerves. Specialized sensory cells, such as this person olfactory epithelium, aggregated polyhedral cells, a small amount, lining of surface of body cavities and granular secretion. Several types of fixed and wandering cells, abundant amounts, support and protect the tissue organs. Elongated contractile cells, moderate amounts. Nerves, elongated cells with extremely fine process, a very small amount, transmission of nerve impulses. Characteristic features of the shape and dimensions of apical cells are quite variable, ranging from tall columnar to cuboidal to low squamous cells. The cell size and the morphology are generally directed by the function. Epithelial cells nuclei vary in shape and may be elliptic, oval, and spherical, or flattened, with nuclear shape corresponding roughly to cell shape. Columnar cells generally have elongated nuclei, squamous cells have flattened nuclei, and cuboidal or palloidal cells have the more spherical nuclei. Because the lipid-rich membrane of epithelial cells are frequently distinguishable by light microscopy, the number and shape of their stained nuclei are important indicators of cell shape and diversity. The nuclei also allow one to determine the number of cell layers in an epithelium, a primary morphologic criterion for classifying epithelia. The nuclei also allow the determination of number of cell layers of the epithelium. Number of cell layers in the epithelium. Most epithelia are adjacent to connective tissue containing blood vessels from which the epithelial cells receive nutrient and O2. Even thick epithelia do not themselves normally contain blood vessels. The connective tissue that underlines the epithelial lining of the organs of the digestive, respiratory, and urinary system is called lamina propria. Lamina propria. The area of contact between the two tissues may be increased by small evaginations called papillae projecting from the connective tissue to the epithelium. Papillae occur most frequently in epithelial tissue subject to friction, such as the covering of the skin of tongue. Projecting from papillae, the area of contractile between the two tissues may increase by small invaginations called papillae. Epithelial cells generally show polarity, with organelles and membrane proteins distributed unevenly within the cells. The region of cell contraction in the ECM, epithelium, cell matrix, and connective tissue is called basal pole. Cell contracting basal pole, and the opposite end, usually forcing a space, is called apical pole, with the two poles differing significantly in both structures and functions. Regions of cuboidal and columnar cells that adjoin neighboring cells comprise of cell's lateral surface. Cell membrane hairs often have numerous folds that increase the area of the functional capacity of the surface. Basement membranes. The basal surface of epithelia rests on a thin extracellular fat-like sheath of micromolecules referred to as the basement membrane. a semi-permeable filter for substances reaching epithelial cell from below. Glycoproteins and other components in this structure can often be stained and visualized with the light microscope. With the transmission of electron microscope, two parts of the basement membrane may be dissolved. Nearest the epithelial cell, the basal lamina, thin, electron-dense, sheath-like layer of fibers, and beneath this layer is a more diffused and fibrous reticular lamina. The term basement membrane and basal lamina sometimes used interchangeably. Basement membrane or basal lamina are sometimes used for interchangeably, but basal lamina usually denotes the fine extracellular layer seen extra the denotes the fine extracellular layer seen ultra ultrastructurally and basement membrane, the entire structure beneath the epithelial cell, visible with the light microscope. The molecules of other basal lamina are secreted from the basal side of the epithelial cell from the sheath-like array. ECM components are described more fully in chapter 5, but those of the basal lamina are characteristically included in the following. Type 4 collagen. Monomers of type 4 collagen self-assemble into two-dimensional network of eventually spread subunits resembling the mesh of a window screen. Lamina. These are large glycoproteins that attach to transmembrane integrin proteins in the basal cell membrane and project through the mesh of formed by typical IV collagen, nidogen or parlectin, respectively a short rod-like protein and proteoglycan. Both of these cross-link lamines to the type 4 collagen network, helping to provide the basal lamina's three-dimensional structure to bind epithelium to the structure and to determine its porosity and the size of molecule able to filter through it. Basal lamina often called external lamina, but with similar composition also exists as a thin slips surrounding muscle cells, nerves, and fat storing cells, where they serve as semi-permeable barriers, regulating micromolecular exchange between the enclosed cell and connective tissue. The more diffuse meshwork of reticular lamina contains type 3 collagen and bound to basal lamina by anchoring basal of type 4 collagen, both of which are produced by cells of connective tissue. Besides acting as filters, functions of basement membranes include helping to provide structural support for epithelial cells and attach epithelia to underlying connective tissue. Basal lamina components help organize integrins and other proteins in plasma membrane of epithelial cells, maintaining cell polarity and helping to localize androcytes, signal transductions and other activities. Basement membrane proteins also mediate many cell-to-cell communications involving epithelia and marks route for certain cell migrations along epithelia. Finally, the basement membrane also serves as a scaffold that allows the rapid epithelium repair and regeneration, intracellular adherence and other junctions. Several membrane-associated structures provide adhesion and communication between cells. Some are present in other tissue, but all are particularly numerous and prominent in epithelia. Epithelial cells adhere strongly to neighboring cells in basal lamina, particularly in epithelial subject to friction and other mechanical forces. As shown on figure 4.4 and summarized in table 4.2, lateral surface epithelial cells have complexes of several specialized intracellular junctions with different functions. Tight or occluding junctions. Junula occluding. both of which are produced by cells of the connective tissue. Tight junction or occluding junction from cell between adjacent cells. Adherent or encroaching junctions are sites for strong cell adhesion. Gap junctions are channels for communication between adjacent cells. In many epithelia, these junctions are present in a definite order at the apical ends of cells. Tight junctions, also called genula occludens, are the most apical of the junctions. The term genula indicates the junction from a bond completely encircling each cell. The term TEM, the adjacent membranes is at these junctions appear fused on every tightly opposed figure 4.5. The seal between the two cells membrane is due to tight interactions between the transmembrane protein claudin and occludin. Tight junctions are clearly seen after cryofracture and epithelia, where they appear as a band of branching strands in the membrane around each cell's apical end. The intracellular seals of tight junctions ensures the molecules crossing an epithelium in either direction, so by doing through the cell intracellular path rather than between them paracellular path. Epithelia with one or very few fused sealing strands, for example proximal renal tube, are more permeable to water and solids than the epithelia with many fused strands, for example a lining of the urinary bladder. Epithelial tight junctions also serve a related purpose. This continuous zone within cell membrane serves as fences, restricting movements of membrane lipids and proteins in apical cell surface into the lateral and basal surface, and vice versa. The tight junction thus maintains two distinct member diamonds, apical and basolateral, with different sets of protein components which allow these two sides of the epithelium to display different receptors and other protein and function differently. The apical cell membrane of epithelia are part of the luminal component of the tissue and organ, while the basolateral domains are part of basal components that also encompasses the underlying connective tissue. Junction: major transmembrane link proteins. Skeletal components. Major functions. Medical reference. Occludins. Claudins. E-adherence. Certain explosives. Cadherence family proteins. Integrins and connexins. Gap junction. Connexins. None. Allow direct transfer of small molecules. Mitosis in various exonin. Medical application. Proteins of tight junctions provide the targets for certain common bacterial and medical importance. The interroxin secreted by the Clostridium preferentiae, which causes food poisoning, binds coding molecules of interstitial cells, prevents insertion of these proteins during maintenance of tight junction, and causes loss of tissue fluid into the interstitial lumen via the paracellular pathway. Similarly, Helicobacter pylori, which is important in the etiology gastric ulcers, binds with extracellular domains of tight junction proteins in cells of the stomach and inserts a protein in the cells, which targets the one and disrupts signaling from junction. The second type of junction is the adherence junction or Jonula adherence. Jonula usually immediately below the tight junction. This is an adherent junction, firmly encroaching to the cell and its neighbors. Cell adhesion is mediated by cadherins, transprotein, glycoprotein, and each cell the bind each with presence of calcium. At their cytoplasmic end, cadherins bind catenins that link to actin filaments with actin binding proteins. This actin filament link to the adherence junction from part of terminal web, a cytoskeletal feature at the apical pole in many epithelial cells. Together, the tight adherent junction encircling the apical ends of the apical cells function like a plastic band that holds six pack of connected rings together. Another enclosing junction is the desmosome, a Marcula adherens. As the name implies, the junction resembles a single spot welt and does not form a belt around the cell. Desmosomes are disc-shaped structures at the surface of one cell are matched with the identical structure at another adjacent cell surface. Desmosomes contain larger membrane of the adherent family called desmogenes and desmocollins. The cytoplasm ends of this clustered transmembrane protein binds plakoglobins, Catherine-like proteins that link to large proteins called desmoplakins in the extron electron dense plate. Desmoplakins in turn bind intermediate filament rather than actins. In epithelial desmosomes attach to fiber-like filaments of cytocatrin, sometimes referred to as tonofilaments. Such intermediate filaments are very strong, and desmosomes provide firm cellular adherence and strength throughout the epithelium. Medical application: various blistering disease such as gap junction, connexomes, hemidesmosomes, focal adhesion kinase, a specialization of apical cell surface, microvilli, brush, strained border, microvilli, esterocili, cilia, 2+9 assembly, axoneme, basal bodies, axonal dynein. Types of epithelia: covering, lining, epithelial and secretory. Covering or lining epithelia. Simple epithelia, stratified epithelia, squamous cuboidal columnar epithelia, stratified squamous keratinized epithelium, keratinization. Major cell functions, examples, distribution and main function. With stratified squamous non-keratinized epithelia, stratified cuboidal, stratified columnar epithelia, transitional epithelium or urothelium, pseudo-stratified columnar epithelium, secretory epithelium and glands, goblet cell, exocrine glands, endocrine glands, secretory portions and ducts. Simple compound, tubular colloid aciniar, branched compound, microcrine secretion, holocrine secretion, apocrine secretion, source and mucor, mucin mucor, seromucous gland, myoepithelial cells. Hormone. transport across epithelia, transcytosis, renewal of epithelial cells, medical application, epithelial tissue, base membrane, intercellular junctions, apical structures, epithelial cells, morphological types of epithelia, epithelial secretion and glands. Cell differentiation in the process by which cells of an embryo becomes specialized structurally to engagement specific cytoplasmic activities or functions at the level of tissue or organs. Organelles are metabolically active structures or complexes with or without membranes in the cytoplasm of eukaryotic cells. Plasma membrane. Plasma membrane or cell membrane or plasma lemma is the lipid bilayer with embedded proteins that surround a cell, and it's seen only in the TEM plasma membrane. The lipid bilayer forms the ampholytic phospholipid stabilized by cholesterol and contains many embedded integral proteins and many peripheral proteins on its cytoplasmic substance. Membrane proteins move laterally within the lipid bilayer, and less movement out in the areas referred to lipid rafts, which have higher concentration of cholesterol and stratified fatty acids. Integral membrane proteins include receptors for external glands, channels for passive and active movements of molecules across the membrane, pumps for active membrane transport, endocytosis and cellular uptake of micromolecules of fluid by plasma membrane, engulfing or invagination, followed by the pinching off of a filled membranous vesicle in the cytoplasm. Major types of endocytosis include phagocytosis, uptake of particular material, pinocytosis, uptake of dissolved substance as a receptors, membrane endocytosis, uptake of specific molecules bound to integral membrane receptor proteins. Exocytosis is a type of cellular excretion in which cytoplasmic membrane vesicles fuse with the plasma membrane fuse with the exocytosis. Membrane vesicles fuse with the plasma membrane to release their contents in the extracellular space. All types of cells similarly use membrane receptors proteins that are often linked to enzymes such as kinase or alanine with those activities initiate intercellular signaling pathways, ribosomes. The two ribosomal subunits, which are complex of rRNA and many proteins attached to mRNA and translate the messenger into protein. Multiple ribosomes on the same mRNA make up polysome, and an abundance of this produces basophilic cytoplasmic and ER staining ribosomes. Of this produce basophilic cytoplasmic staining endoplasmic reticulum. The ER is a covalent network of membrane enclosing continuous space called cisternae and extend from the nucleus to the endoplasmic reticulum to a plasma membrane. Rough endoplasmic reticulum is granular basophilic cytoplasmic substance due to the presence of polymers, making most membrane proteins in certain other organelles or exocytosis. RER is always well developed in cells actively secreting proteins. Proteins to be processed through the RER contain initial single peptides, bind receptors in the ER membrane, localize them to granulae after translocation across the membrane into the cisterna. The proteins undergo post-translational modification and folding in the process monitored by RER molecular champions and enzymes. Smooth ER lacks ribosomes but includes ribosomes for lipid and glycogen metabolism, for deoxyribonucleation reactions, and for temporary separations. Golgi apparatus. The Golgi apparatus is a dynamic granule considering the stuffed membrane system in which proteins made in RER are processed further and packaged for secretion and other roles. Proteins in transport vesicles, either on the cis or receiving trace on the Golgi, move through the medial cisternae Christie, mitochondria, cysteine, Golgi body, network, enzyme, and released in the other vesicle in a transphase. Vesicles movement through the Golgi apparatus. Golgi apparatus is guided by specific code proteins such as COPII and COPI. Important protein modification Golgi apparatus includes sulfur of many glycosylation reaction. Modified proteins leave the Golgi apparatus after packaging in vesicles and other proteins that direct movement of the lysosome, the plasma membrane, or secretion by exocytosis. Primary lysosomes emerge from the Golgi apparatus containing inactive acid hydrolysis specific for degrading a wide variety of cellular molecules. Secondary lysosomes are more heterogeneous, having fused with vesicles produced by endocytosis that contain material to be digested by the hydrolytic enzymes. During autophagy, lysosomes digest unneeded or non-functional organelles after this surrounded by membrane that then fuses with the lysosome. Products of digestion in secondary lysosome are released to the cytoplasm for reuse. Final condensed vesicles containing any indigestible molecules are called residual bodies. Proteosomes. Proteosomes are small cytoplasmic protein complexes that degrade improperly folded proteins that they are tagged with the polypeptide ubiquitin. Proteosomes, they are tagged with the polypeptide ubiquitin. Mitochondria. Mitochondria are the major site of ATP synthesis and abundant cell cytoplasmic regions where large amount of energy are expended. Mitochondria are usually elongated organelles that form fission of pre-existing mitochondria. Mitochondria have two membranes, a porous outer membrane encloses the inner membrane space and inner membrane with many pores, like gel-like matrix. The mitochondrial matrix contains enzymes for deoxidation of fatty acids and cristaceous Krebs cycle. The inner membrane includes enzymes assembled in electrotransport system and ATP synthesis. Mitochondria is of stress. A cell may release cytochrome C from the inner membrane, triggering the regulating series of accumulating of cell death. Cytochrome C. Mitochondria stress may release cytochrome C of the inner membrane cell apoptosis. Peroxisomes are small spherical organelles containing enzymes for various metabolic reactions, notably Paroxysm is small spherical organelles containing enzymes for various metabolic reactions, notably for oxidation and detoxification, and catalytic that breaks down H2O2 resulting from these reactions. Cytoskeleton. The cytoskeleton contains three types of polymers: microtubules, 25 nanometer diameter, and actin filaments, and intermediate filaments. Microtubules have semi-rigid tubular structures. Resulting from the cytoskeleton contains three types of polymers: microtubules, 5 nanometer. Microtubules are semi-rigid tubular structures and walls composed of polymerized tubulin. Structure where dynamic and steady addition to dissociation of tubulin. Microtubulins. Ketone and dynamic tubes are important maintaining cell shape tracks for transport of vesicles. Organelles, motor, protein, ketones, and amine. Microfilaments are short, flexible, dynamic filaments. Actin subunits, banded proteins, myosin and protoproteins that bind with filaments. Movements of cytoplasm produced by actin filaments during mitosis are important for endocytosis. The cell cleavage after mitosis and co-motion of substance. Intermediate filaments are the most stable cytoplasmal components, according to strong mechanical stability to cell. Intermediate filaments are composed of various products and supplements in different cells. These include T and lamins. This uses different cells. Gametes. Vimentin and lamins. Neurofilaments. Keratin, which is especially important for epithelial cells, includes unlike organelles, includes are not metabolically active and are primary storage cells such as lipids, droplets, glycogen granules, pigment granules, and residual bodies. Also, lipofuscin. Chapter 3 Nucleus has components of the nucleus, nuclear envelope, chromatin. Components of nucleus, chromatin, nuclear envelope, nuclear envelope, perinuclear space, nuclear lemma, lemmis, nuclear pore. Examples: nucleoporins. Importance: chromatin, chromosomes, chromatids, histones, nucleosomes, achromatin, heterochromatin, centromeres, telomeres, bar body. Chromosomal territories: homologous, diploid, haploid, karyotype. Nucleus, and then the cell cycle, mitosis, G1 phase, S phase, G2 phase, G0 phase, mitogens, growth factors, as cyclic dependent kinase, CDKs, G1 restriction point. Then there is mitosis, interphase, prophase, mitotic spindle, metaphase, kinetochromes, anaphase, telophase, cytokinesis. There is cyclin D, cyclin D6, cyclin E, cyclin D2, DEAAB. And there is stem cell and tissue renewal, stem cells, progenitor cells, meiosis, gametes, synapses, crossovers, haploids, and then apoptosis, BCL2 family, loss of microtubule function and caspase activation, fragmentation of DNA and the nucleus, shrinkage of nuclear and cell volumes, pyknotic nuclei, cell membrane changes, formation and phagocytic removal, apoptotic bodies. Medial application. Intracellular connections. List of the intracellular junctions and connections. Describe any two of them. Intracellular junctions, intracellular adhesions, junction complex. Several membrane associated structures contribute to adhesion and communication between cells. These structures are collectively called junctional complex. They are present in most tissues, are particularly numerous prominent in epithelia. Types: occluding impermeable junctions, seals to prevent the flow of materials between the cells. Tight junctions or Jonule occludents, single Jonula occludents. Adhesive or anchoring or adhesing junctions, sites of adhesion. Jonula adherens, desmosome or muscular adherens. Jonula adherens, muscular adherens. Desmosomes and hemidesmosomes. Communicating junctions, channels for communication between the adjacent cells, gap junction. Short node gap junctions. Definition: The junction by which molecules are transferred from one cell to another is called gap junctions. Distribution: epithelial tissue, cardiac muscle cells, smooth muscle cells, and neurons. Distribution: gap junctions are epithelial tissue, cardiac muscles, smooth muscles, and neurons. Structure: at gap junction, the intracellular space narrows from 20 nanometer to 3 nanometer, 35 nanometer to 3 nanometer in gap junctions. Gap junctions. The units of gap junctions are called connections. Connections. Each connection is made out of 6 protein subunits called connexins. When connections are adjacent, cells are limited up with one another. They form a channel that connects the cytoplasm of those two cells. The diameter of the channel is normally about 2 nanometer. Power function. Gap junctions permit substance to pass between the cell without entering the ECF or intracellular fluid. Permits the passage of ions, sugars, amino acids, and other solutes with molecular weight up to about 1000. It helps in exchange of chemical messenger between the cells. It helps in rapid propagation of action potential from one cell to another cell. Intracellular communications. Briefly describe the intracellular communications examples of each. Cellular communications. Definition: it is a sophisticated communication system to coordinate with functions of cells, tissues, and organs by a variety of regulation signals to maintain the internal environment of the maintenance of life. Mechanism: cells communicate with one another via chemical messenger. With a given tissue, some chemical messenger secrete into the intracellular fluid by direct cell-to-cell connects. Chemical messengers typically bind to proteins receptors on the surface of the cell, or in some instances in cytoplasm or in the nucleus, delegating sequence of intracellular changes that produce their physiologic effects. Types of intracellular communications. Chemical messenger systems. Types: definition and chemical messenger. Neurocrines. Here, neurotransmitters are released at the sympathetic junction and act locally to control cell function. Neurotransmitters. Endocrine. Here, glands or specialized cells release hormones in the blood that influence the function of the cells at another location of the body. Hormones. Neuroendocrine. Here, neurons secrete neurohormones that reach the circulating blood and influence the function of cells at another location in the body. Neurohormones: ADH, oxytocin. So, neurocrine, endocrine, and neuroendocrine. Paracrine. Here, cells secrete substances that diffuse into the ECF and affect neighboring cells. Cytokinesis, eicosanoids, platelet activating factor, vasoactive substance, histamine, serotonin, and bradykinin. And autocrine are here cells secrete substances that affect the function of the same cell by binding to the cell surface receptors. Cytokinesis, VAS. So, neurocrine, neurocrine, endocrine, neuroendocrine, paracrine, and autocrine. Neurocrine are neurons, and neurocrine are neurotransmitters. Endocrine are hormones. Neuroendocrines are neurohormones. Neuroendocrine. And paracrine are cell substances diffuse with ECF and affect neighboring cells. paracrine, and autocrine are cell substrates that affect the function of the same cell by binding with the cell substrate receptors. Comparison between intracellular communications. Gap junctions, neurocrine, synaptic, paracrine, autocrine, and endocrine. Messenger transmission. Local or general. Specific depends on. Gap junctions. Directly from cell to cell. Gap junctions. Local or general. Local. Special depends on automatic location. Neurocrine. Across sympathetic cleft. Local. Anatomic location and receptors. Paracrine and autocrine. By diffusion in the interstitial fluid, locally diffuse and receptors. Endocrine by circulating body fluids, general and receptors. Gap junction, synaptic, paracrine and autocrine and endocrine. Intercellular communications by chemical mediators, autocrine and paracrine. Gap junction, synaptic, paracrine and autocrine, and endocrine. Membrane transport. What is membrane transport? Membrane transport definition. It is a biophysiochemical phenomenon by which different substances are transported across the cell membrane from inside and outside the cell, vice versa. Membrane transport. Active transport, passive transport. The passive transport can be divided into osmosis and diffusion. Diffusion further on can be divided into simple diffusion and facilitated diffusion. Osmosis, diffusion, simple diffusion, facilitated diffusion. Active transport. Primary active and secondary active. Primary active transport includes sodium, potassium pump, calcium pump, and hydrogen pump, etc. Secondary active transport includes co-transport and counter-transport. Co-transport, sodium glucose co-transport and sodium amino acid co-transport. Counter-transport includes sodium calcium counter-transport and sodium hydrogen counter-transport. Transport across macromolecules. So membrane micromolecules are active, passive, and diffusion, simple diffusion and facilitated diffusion. Active, primary active, secondary active. Primary active, secondary active. Secondary also includes co-transport and counter-transport. Co-transport and counter-transport. Co-transport includes sodium glucose and sodium amino. Counter-transport includes sodium calcium and sodium hydrogen. Exocytosis. These are macromolecules. Exocytosis. Neurotransmitters, hormones, enzymes, proteins are released from the cell by this process. Exocytosis. Neurotransmitters, hormones, enzymes, proteins are released. Endocytosis are two types: pinocytosis and phagocytosis. Pinocytosis, for example, ingestion of protein, vitamin, etc. Pinocytosis. Phagocytosis, ingestion of bacteria, dead cell and tissue debris, etc. Carrier proteins, channel proteins. Simple diffusion, facilitated diffusion, energy. Carrier proteins. Diffusion, active transport. What do you mean by carrier mediated transport? Carrier mediated transport process with examples. Carrier mediated transport involves a specific protein carriers transport that bind with and move molecules across the membrane. These processes are selective, saturable, and regulated. Carrier mediated transport process with examples. Carrier mediated transport process involves a specific protein carriers transports that bind and move molecules across the membrane. These processes are selective, saturable, and regulated. Transport type, mechanism, energy requirement, and example. Facilitated diffusion. Passive, moves solution down gradient via carrier proteins. Down gradient. No ATP, passive. GLUT or GLUT4 transports for glucose. Primary active transport. Direct ATP used to ATP. Pump solute against gradient. Down gradient against gradient. Primary actors ATP dependent. ATP is secondary active transport. Uses ion gradient to drive solute transport. Indirect ion gradient. SL SGLTI. Glucose cotransport. Antiport. Hydrogen ion exchange. Ion channels. Gated ports. Passive. Voltage gated sodium channels. Ion channels. Key features of carrier mediated transport. Specificity. Each carrier binds particular molecules, for example, glues of glucose. Structurability. Limited by the number of carriers. Number of carriers follows Michaelis Menten kinesis. Regulation. Hormones, for example, insulin and upregulate. Clinical relevance. Diabetes. Impaired GLUT4 translocation. Insulin resistance. Cystic fibrosis. Defective CFTR. Thick mucus. Digoxin toxicity. Inhibits sodium potassium ATPase pump. Clinical correlation involves diabetes, cystic fibrosis, and digoxin toxicity. Active transport. Active transport definition. The movement of substance across the cell membrane against chemical or electrical gradient with the active expenditure of energy by the help of carrier protein is called active transport. Definition. Active transport means movement of ions or other substances across the membrane in combination with a carrier protein against a concentration gradient. For example, low concentration to high concentration state. Concentration of the solute and the solvent. Concentration of the solute. Definition. Active transport means movement of ions or other substances across the membrane in combination with a carrier protein against a concentration gradient, i.e. the low concentration to high concentration state, or against an electrical or pressure gradient with the active expenditure of energy. Definition. Characteristics. Criteria of active transport. Primary active transport. The key criteria for active transport are movement against a concentration gradient, required energy, involvement of carrier proteins, selective and saturable, and regulation by cellular needs. Movement against a concentration gradient. Substances are transported from an area of low concentration to higher concentration. Uphill movement, unlike passive movement, requires energy. Primary active transport directly uses ATP. Secondary active transport uses energy from the electrochemical gradient created by the primary transport. Electrochemical gradient created by the primary active transport. Involvement of carrier proteins. Specific membrane proteins, pumps, symports, and antiporters bind and transport molecule ions selectively. Selective and saturable. Only specific molecules are transported. Highly specific. The process can reach a maximum rate when all transports are occupied. Saturation kinetics. Regulation by cellular needs. Active transport is tightly controlled by cellular signals, for example, hormones, phosphorylations, and meet metabolic demands. Classification. It is of two types. Primary active transport. The movement of substance across the membrane against the chemical or electrical gradient with the active expenditure of energy which is derived from ATP or some other high energy phosphate by the help of carrier protein is called primary active transport. Among the substances that are transported by primary active transport are sodium, potassium, calcium, hydrogen, chlorine, and a few ions. For example, sodium potassium pump, calcium pump across the membrane against chemical or electrical gradient. Against the chemical or electrical gradient. Electrical gradient with the active expenditure of energy. High energy phosphate, active of ATP or some other high energy phosphate by the help of carrier proteins. Hydrogen pump, etc. Secondary active transport, the movement of substances across the cell membrane against chemical or electrical gradient with the energy which is derived secondly from the concentration of electron gradient produced by primary active transport by the help of carrier protein is called secondary active transport. It is again of two types. Secondary active, the movement of substances across the cell membrane against chemical or electrical gradient with the energy which is derived from concentration of electrical gradient produced by primary active transport by the help of carrier protein is called secondary active transport. It is again of two types: co-transport, symport, and counter-transport, antiport, symport. Secondary active transport of two substances in the same direction is called secondary active co-transport. For example, sodium glucose co-transport. Counter-transport, secondary active transport of two substances in the opposite direction is called secondary active or counter-transport. For example, sodium hydrogen counter-transport. Mechanism of primary active transport, substances to be transported plus ATP bind to their respective sites in the carrier protein. Breakdown of ATP and ADP and phosphate. Phosphate binds to the phosphorylation site of the carrier protein. Conformation changes of the carrier protein. Efflux or influx of the substance. So ATP bind to their respective site and carrier protein. Breakdown of ATP and ADP. Phosphate binds with phosphorylation site of the carrier protein. Formation of change of the carrier protein and efflux or influx of substances. Mechanisms of primary active transport. Substance to be transported plus ATP bind to their respective sites in the carrier protein. Breakdown of ATP and ADP. Phosphate binds with the phosphorylation site of the carrier protein. Conformation changes in the carrier protein, efflux and influx of substances. Mechanism of secondary active transport. Firstly, inwardly direction of sodium gradient is produced. ECF sodium is greater than ICF sodium. Extracellular sodium is greater than the intercellular sodium. By primary active transport of sodium pump. The primary active transport of sodium pump. The gradient represents a storehouse of energy because the excess of ECF sodium is always attempted to diffuse in the interior. When both sodium and second substrate bind to the outer portion of the carrier protein, conformation changes occur in the carrier protein and both sodium and the second substance move to the interior of the cell. Secondary active transport. For example, sodium glucose co-transport. When sodium binds to the outer portion of the second substrate, binds into the inner portion. When sodium binds to the outer portion of the second substrate, and the second substrate binds to the inner portion of the carrier protein, conformation changes occur in the carrier protein and sodium is transported into the interior of the second substance is transported to the exterior of the cell. Exterior of the cell, and sodium is transported into the interior of the cell. Secondary active counter transport. Sodium hydrogen or sodium calcium counter transport. Mechanism of secondary co-transport is sodium glucose and co-transport. Gives the basic mechanism of transport and sodium ions across an epithelial sheath with diagram. Gives a basic mechanism of transport of sodium ions across epithelial sheath with diagram. Basic mechanism for transport of sodium ions across epithelial sheath. Secondary active co-transport. For example, sodium glucose co-transport. Secondary active counter transport. Sodium hydrogen sodium calcium counter transport. How does sodium ion help in co-transport and counter transport? With example. Role of sodium ion in co-transport and counter transport. When sodium ions are transported out of the cell by primary active transport, a large concentration gradient of sodium usually develops. Very high concentration outside the cell and a very low concentration inside. This gradient represents a storehouse of energy because the excess of sodium outside and the cell membrane is always attempting to diffuse in the inferior. Under appropriate conditions, this diffusion energy of sodium laterally can pull other substances along with the sodium through the cell membrane. Role of sodium ion in co-transport. Glucose and amino acids are transported in most cells against large concentration gradient. Glucose and amino acids are transported in most cells against large concentration gradient by co-transport with sodium. Glucose and amino acids are transported into most cell against large concentration gradient in co-transport with sodium. During sodium glucose co-transport, when both sodium and glucose bind to the carrier, when both sodium and glucose binds to the carrier protein, a conformation change occurs in the carrier protein and the sodium and glucose are transported to the outside of the cell at the same time. Bind to the carrier protein, a conformational change occurs in the carrier protein and sodium and glucose are transported to the inside of the cell at the same time. Sodium amino acid co-transport occurs in the same manner as for glucose. Sodium amino acid co-transport. Role of sodium ion in counter transport. During calcium calcium counter transport, both sodium and calcium ion bind to the carrier protein and a conformational change occurs in the carrier protein and the sodium ions move to the interior and calcium ions move to the exterior. Sodium hydrogen counter transport occurs in the same manner. Some useful terms. Nice to know. Uniports. A uniport is a transport protein which transports only one substance. Symports. Co-transports. These transport proteins transport more than one substance across the membrane together. For example, a symport is the interstitial mucosa causes co-transport by facilitated diffusion of sodium and glucose from the interstitial lumen into the mucosal cell. Co-transport is a transport of protein transport more than one substance across the membrane together. And antiports. Antiports are transport proteins that exchange one substance for another. For example, sodium and potassium. Filtration. It may be defined as the process by which fluid is forced through the membrane or other barrier because of the difference in pressure on the two sides. The capillary wall separating the plasma from the interstitial fluid differs across it make filtration a significant factor in producing movement of water and solute. Ultrafiltration. It is the process by which small crystalloid molecules are separated through a filtering membrane. Such filtration takes place in the separation of plasma protein from other plasma solutes in the glomerular membrane of kidney during formation of urine. Ultrafiltration. Sodium potassium pump or sodium potassium ATPase. Definition: the sodium potassium pump is a primary active transport mechanism which pumps 3 sodium from inside and 2 outside at the same time, 2 potassium from the outside to the inside of the cell across the cell membrane against their concentration gradient with active expenditure of energy from hydrolysis of ATP by the help of carrier protein. The structure of sodium potassium pump. Carrier protein for sodium potassium pump has alpha and beta subunit. Inner portion of alpha subunit has 3 binding sites for sodium, 1 binding site for ATP and 1 for phosphorylation. Carrier protein for sodium potassium pump has alpha and beta subunit. Inner portion of alpha subunit has 3 binding sites for sodium and 1 binding site for ATP, 1 site for phosphorylation. Mechanism for sodium potassium pump. Sodium potassium or ATP bind to their respective site for alpha subunit of the carrier protein. Breakdown of ATP and ADP phosphate. Phosphates bind to the phosphorylation site of alpha subunit of the carrier protein. Conformation site of the carrier protein. Efflux of sodium and influx of potassium. Efflux of 3 sodium and influx of 3 potassium. Important: physiological. It is impossible for generation of membrane potential. It controls cell volume. Physiological. It is responsible for the generation of membrane potential. It controls cell volume. It is the basic of nerve and muscle function. It provides energy for secondary active transport. Clinical. Sodium potassium pump digital is used for heart failure. Dopamine in the kidneys inhibits sodium potassium pump, causing natriuresis. Control of cell volume by sodium potassium pump. Control of cell volume of sodium potassium pump. The sodium potassium ATPase pump always a crucial role in regulating cell volume by maintaining osmotic balance and preventing excessive water influx. Here's how it works. Sodium potassium pump sodium 3 sodium ions out and 2 potassium pairs ATP hydrolyzed and creates a electrochemical gradient, high extracellular sodium and high intracellular potassium. Role of cell volume regulation. Preventing cell swelling. Without the pump, intracellular sodium would accumulate, increasing osmotic pressure. Water would enter the cell via osmosis, causing cell swelling and potential lysis. By promoting out sodium, the pump reduces intracellular solute concentration, balancing osmotic pressure. Compensation for water movement. Cell constantly face exchanges in extracellular solution contractions. If extracellular osmolality decreases, hypotonic solution water tends to rush in. The sodium potassium pump helps in expel sodium, reducing intracellular osmolarity and countering swelling. Secondary role of sodium and chloride channels. Sodium ions pumped in the sodium potassium ATPase can exit via potassium leak channels. Balancing charge. Chlorine often follows sodium passively, maintaining intracellularity. This sodium chloride efflux further regulates cell volume by reducing intracellular solutes. Failure of the pump leads to cell swelling. If the secondary role of potassium and chloride channels. Potassium ion pumped in by the sodium potassium pump can exit via potassium leak channels, balancing charge. Failure of the pump leads to cell swelling. If the sodium potassium pump inhibits, for example by iobane or hypoxia, sodium is inhibited by iobane or hypoxia, sodium accumulates inside the cell. Water follows cell swelling. by Oyudian. In extreme cases, it leads to cytolysis, cell bursting. Nice to know. Electroneural transport occurs when the net electrical charge remains balanced during transport. For example, during sodium antiport and calcium and sodium chlorine symport. What is electrogenic pump? Electrogenic pump, the pump which causes net change transport across the membrane and thus creates a electric potential across the cell membrane is called an electrogenic pump. Or the electrogenic pump is a membrane transport potential that generates a electrical gradient membrane protein by moving net charge across the cell membrane. Unlike electroneural pumps which exchange equal charges, electrogenic pumps create an imbalance in charge distribution, contributing to the cell's resting membrane potential. What is electrogenic pump? Definition: the pump which causes net charge transport across the membrane and thus creates an electric potential across the cell membrane is called an electrogenic pump. The pump which causes net charge transport across the membrane and thus creates an electric potential across the cell membrane is called an electrogenic pump. Or the electrogenic pump is a membrane transport protein that generates an electrical gradient membrane potential by moving net charge across the cell membrane. Unlike electroneutral pumps which exchange equal charges, electrogenic pumps create an imbalance in charge distribution, contributing to the cell's resting membrane potential. Key features of electrogenic pumps: net movement of charge, transport ions in equal ratios leading to a separation of charges across the membrane. Example: the sodium potassium ATPase pump 3 and sodium out for every 2 potassium ions in, creating a net outward positive movement. Contribution to membrane potential: helps establish the maintain a negative resting potential inside the cell, typically minus 70 millivolts in neurons. Energy source: most electrogenic pumps are primary active transports, use ATP directly, and some are secondary transports, hydrogen coupled symports with plants or fungi. Examples of electrogenic pumps: pump type, action and role. Pump type: sodium potassium pump, 3 sodium and 2 potassium, maintains resting potential cell volume. Hydrogen ATPase. Proton pump, pumps hydrogen, for example stomach and lysosomes, acidifies compartments and drives secondary transport. Calcium ATPase pumps calcium out per ATP, muscle relaxation, stimulating termination. Biological importance: neuron and muscle function. Sodium potassium ATP is a critical for action potential and muscle contractions. Nutrient absorption: the hydrogen gradient created by electrogenic pump drives uptake of nutrients, for example glucose and antioxidants. Phosphate pH regulation. Proton pumps in kidneys, maintaining acid-base balance. Electrogenic versus electroneutral pumps. Features: charge movement, example effect on potential. Electrogenic pump creates net charge balance. Sodium potassium ATPase directly alters membrane potential. Electroneural pump, no net charge movement, and sodium hydrogen exchange, no direct effect of potential. Why sodium potential pump is called electrogenic pump? Sodium potassium pump is called the electrogenic. The sodium potassium pump is said to be an electrogenic pump because it generates an electrical potential voltage difference across the cell membrane, creating a net movement of charge. The sodium potassium pump moves 3 sodium ions in the exterior for every potassium ion in the interior. As a result, the net positive charge is moved from the interior in the cell and exterior outside of the cycle of the pump. This creates positivity outside of the cell and negativity inside the cell. Thus, an electric potential across the cell membrane is created. Therefore, the sodium potassium pump is said to be electrogenic. The state and the function of sodium potassium pump, significance, clinical importance and relation and differences with the secretory active transport. Function, physiological significance and clinical importance. Relation with the secondary active transport. Sodium potassium pump produces an inwardly direction of sodium gradient, which act as energy for secondary active transport. Difference between sodium potassium pump and secondary active transports. Features: sodium potassium pump and secondary active transport. Energy source: directly uses ATP hydrolysis, less uses pre-existing ion gradient, hydrogen or sodium created by primary pumps. Pumps 3 sodium and 2 potassium in per ATP. Couples downhill movement of one solute, for example sodium, and uphill transport of another, for example glucose. Role in gradient: creates ion gradient, high extracellular sodium, high intracellular potassium. Exploits gradients, does not create them. Transports proteins, ATPase, and sympaths, antipodes. Electrogenic S. Net charge movement contributes to the membrane potential. Depends. Sympaths are electrogenic. Antipodes are electroneutral. Examples: sodium potassium ATPase, hydrogen ATPase. Sympaths: sodium glucose transport. Antipodes: sodium potassium exchange. Biological role: maintains resting potential, cell volume and osmotic balance, absorbs nutrients, glucose, amino acids, and removes waste. What is sick cell syndrome? Sick cell syndrome, also called cell volume dysregulation syndrome, refers to a pathological cause of sickle cell syndrome: energy deprivation, ischemia, hypoxia, mitochondria dysfunction, toxin exposure, Quabian and digoxin. Alcohol, heavy metal, Quabian, dioxin. Ischemia, hypoxia. Disease states: chronic kidney disease and diabetes. Neurodegenerative disorders, Alzheimer's, where neural sodium and potassium pump actively declines. Key symptoms and effects: cell shrinkage and cell swelling. Cell accumulates inside, water influx due to the osmotic imbalance. Potassium leaks out, organelles dysfunction. Apoptosis, necrosis, cell shrink, apoptosis and necrosis. Clinical implications: brain cells, neuronal shrinkage, cognitive decline, muscle cells, impaired sodium potassium pump, fatigue, cramps. Kidney cells, dysregulated volume, hypertension, edema. Reference: mechanism and significance of cell volume regulation. Cell sickness syndrome, CSS. The term cell sickness syndrome is not formally recognized medical or scientific diagnosis, but is occasionally used in broader context to describe a state of chronic cellular dysfunction caused by cumulative states. what cell sickness syndrome implies. Generalized cellular dysfunction. Cells struggle to maintain homeostasis due to oxidative stress. Mitochondrial dysfunction, toxin accumulation, chronic inflammation. Proposed symptoms: fatigue, muscle weakness, accelerated aging, and increased susceptibility to disease. How cell sickness syndrome (CSS) differs from sick cell syndrome. How cell sickness syndrome (CSS) differs from sick cell syndrome. Definition: information term for global stress, formal condition for sodium potassium pump failure. Scope: vague, holistic, specific, scientific basis, lacks peer reviewed diagnosis criteria, well documented in physiologic literature. Terminate focus: detox, antioxidants, lifestyle changes, carrier arms, and transport. Discuss the process of glucose movement across the cell membrane with diagram. Process of glucose movement across the cell membrane. Sodium glucose co-transport. This occurs in the brush border of interstitial epithelium and luminal part of the renal tubular cell membrane through sodium dependent glucose transport. Sodium dependent glucose transport CGLT facilitate diffusion. This occurs in the basolateral part of interstitial epithelium and renal tubular cell membrane through glucose transport GLUT, and insulin dependent transport in muscles and most cells of the body with few exceptions. Diffusion. Diffusion may be defined as the movement of solute particles by the normal kinetic motion from a higher concentration to a lower concentration. Example: exchange of oxygen and carbon dioxide in lungs and tissue occurs by diffusion. Types: two types. Simple diffusion does not require carrier protein, for example simple diffusion O2 and CO2, etc. Facilitated diffusion requires carrier protein, facilitated diffusion of glucose. Factors affecting rate of diffusion: concentration difference. Rate of diffusion is similar to concentration difference. Equivalent to cross section area. Rate of diffusion equivalent to cross section of area corresponds to diffusion may be defined as the movement of solute particles by the normal kinetic motion from a higher concentration to a lower concentration. Exchange of O2 and CO2 in lungs and tissues occur by diffusion. Simple diffusion does not require carrier protein, for example simple diffusion O2 and CO2, etc. Facilitated diffusion requires carrier protein, for example facilitated diffusion of glucose. Factors affecting rate of diffusion: concentration difference, cross section area, distance between two regions, temperature and size of particles, molecular weight of particles, thickness of the cell membrane, lipid solubility, number of protein channels in the cell membrane. Importance of diffusion: exchange of O2 and CO2 in lungs and tissue occurs by diffusion. Admixture of foodstuffs and indigestion juices occur by diffusion. Absorption of pentose, minerals, water, insoluble vitamins, etc. GIT occurs by diffusion. Secretion of waste products NH3 into renal tubules occur by diffusion. Diffusion of sodium and potassium through sodium-potassium leak channels create resting membrane potential. Exchange of capillary bed occurs by diffusion. Pneumonia, thickness of alveoli membrane Diffusion, thickness of alveolar membrane, pneumonia. Diffusion of O2, hypoxia. How diffusion occurs through the cell membrane. How diffusion occurs through the cell membrane. Simple diffusion. Through simple diffusion. Through the interstices of the lipid bilayer of diffusion substrate is lipid soluble. For instance, the lipid solubility of oxygen, nitrogen, and carbon dioxide and alcohol are high, so that all these can dissolve directly in the lipid bilayer and diffuse through the cell membrane. Through watery channels, the penetrate all the way through some of the large transport proteins. Facilitated diffusions. Facilitated diffusions requires interactions of carrier protein. The carrier protein aids passage of the molecules of ions through the membrane by binding chemically with them and shuttling them through the membrane in this form. What is facilitated diffusion and why it is called facilitated diffusion? It is a passive transport process where molecules move down their concentration gradient high to low concentration gradient across the membrane with the help of specific carrier protein channels. Unlike simple diffusion, it requires membrane proteins to assist the movement of molecules so that large or polar or cross a lipid bear freely. It is called facilitated diffusion. It is called facilitated because protein mediated molecules rely on transport proteins to pass through the membrane. Example, GLUT transports facilitate glucose entry into cells. No energy ATP required. Movement of driven solely by the concentration gradient, like simple diffusion, not protein facilitated process. Overcome membrane barriers. Membrane transports of molecules that cannot diffuse freely. For example, iron, glucose, amino acids. Examples: glucose transport GLUT4, insulin regulated in muscle fat cells, ion channels, aquaporins, water channels, aquaporins, sodium leak channels, maintain resting membrane potential. Clinical relevance: diabetes, impaired with GLUTA function, insulin resistance, cystic fibrosis, defective CFTR channel, thick mucus, state with fixed law of diffusion. Fixed law of diffusion: the magnitude of the diffusion capacity from one region to another region directly proportional to the cross-section area across which diffusion is taking place, and concentration gradient or chemical gradient, which is different in concentration of the diffusion substance divided by the thickness of the boundary. Thus, J is equal to minus dA dC dx, and J is the net rate of diffusion, D is diffusion coefficient, A is the area, dC dx is the concentration gradient. Osmosis. Definition: the diffusion of solvent molecules across a semi-permeable membrane from the region of lower solute concentration to the region of higher solute concentration is called osmosis. Condition of osmosis: two solutions of different osmotic pressure. Presence of a semi-permeable membrane between two solutions. Importance of osmosis. Physiological: absorption of GIT, fluid exchange in capillary bed, regulation of urine formation, reabsorption of CSF, regulation of water balance in the body, maintenance of blood pressure. Clinical: Infusion of isotonic solution 0.1 sodium chloride 5% glucose solution to maintain blood volume in the dehydration burn. In cerebral edema, important osmosis. Cerebral edema, hypotonic solution is used, which causes osmosis of water from the brain and thus reduced edema. Volume dehydration, maintain blood volume dehydration in burn. 0.9% HCl 5% glucose solution. The cerebral edema, when plasma proteins concentration is decreased, water leaves the blood vessel and enters into the interstitial space by osmosis. Thus, edema develops. The RBC are taken in a hypotonic solution. Water enters into the RBC by osmosis. RBC swells and eventually bursts. Hemolysis. Purgatives prevent water absorption by osmosis from GIT. Thus, they cause dilution of interstitial contents and help in defecation. GIT, thus they cause absorption of osmosis from GIT. Prevent water absorption from osmosis from GIT. Osmotic pressure, the amount of pressure required to prevent the osmosis is called the osmotic pressure. Factors regulating osmotic pressure. In an ideal solution, osmotic pressure P is related to temperature, T and volume in the same way as the pressure of gas. P equal to nRT by V. Number of osmotically active particles are equal to gas constant. From this equation, we can find the factors. Number of osmotically active particles. Osmotic pressure is directly proportional to the number of osmotically active particles present in the per unit volume of solution. Temperature, OP is directly proportional to the temperature. Volume, OP is inversely proportional to the volume of the solution. Effect and ionization. Number of ionization. Ionizable solutes have more osmotic pressure. Osmotic pressure is proportional to the volume of the solution, is inversely proportional to the volume of the solution. Osmotic pressure is proportional to the temperature. Osmotic pressure is proportional to the volume per unit. Effect of ionization. Ionization solutes have more osmotic pressure because ionizable solutes dissociate into two or more ions. Each of the ions exert OP individually, ions of the solute, and affect the aggregation. Some substances from molecular aggregation in solution. This decreases number of osmotically active particles. Start note: colloidal osmotic pressure. Colloidal osmotic pressure, the osmotic pressure that is extended by the colloid is called colloidal osmotic pressure. Volume of colloidal osmotic pressure. Colloids exert a low osmotic pressure. Plasma proteins exert osmotic pressure of about 28 to 32 nanometers mercury, average 30 parameter mercury. Importance: in the capillary bed, plasma colloidal osmotic pressure causes osmosis of tissue fluid from the interstitial space back to the blood vessels. In capillary bed, plasma colloidal osmotic pressure causes osmosis of tissue fluid from interstitial space back to the blood vessels. Interstitial space back to the blood vessels. Plasma proteins exert colloidal osmotic pressure which retains plasma in blood vessels, thus maintaining plasma volume. Intercellular proteins exert colloidal osmotic pressure, which controls intracellular fluid volume. Protein in the intracellular space exerts colloidal osmotic pressure, which controls interstitial fluid volume. It plays important role in regulation of glomerular filtration. Discuss the role of osmotic pressure and the importance of fluid volume. Role of osmotic pressure in maintenance of fluid volume. Osmotic pressure is a critical force that regulates fluid balance across the cell membrane and blood vessel by driving water movement in movement. It ensures proper fluid distribution between intracellular and extracellular compartments, maintenance of homeostasis. Osmotic pressure is the pressure required to prevent water movement across the semi-permeable membrane due to solute concentration difference. It is governed by osmolarity, total solute particle L, and tonicity. Osmotic pressure gradient. Key roles in fluid volume maintenance. Intracellular fluid ICF balance, cell volume stability, high ECF osmolarity, low ECF osmolarity. Water leaves the cell, water enters the cell. Cell swelling, cell shrinkage. Regulated by sodium potassium ATPase pump and aquaporins. Regulated by aquaporins. Extracellular fluid balance, plasma volume, albumin generates colloid osmotic pressure and retain water in capillaries. Low COP, nephrotic syndrome, edema, interstitial fluid balance by Stirling forces, hydrostatic versus osmotic pressure, renal water reabsorption, kidney adjusts water reabsorption based on osmotic gradients. ADH, increase absorption to channels in collection ducts, water retention. Counter current multiplier creates medullary osmotic gradient for urine concentration. Which one will exert more osmotic pressure, colloid or crystalloids? Once the size of the colloid particles is larger than the size of the colloid molecules, the number of colloid particles is less relative to crystalloid molecules in some amount, as osmotic pressure is directly proportional to the number, not the size of the particle. Colloid exerts less osmotic pressure than crystalloids. Which one will exert more osmotic pressure, glycogen or glucose? Glucose is a crystalloid, but glycose is colloid. Glucose exerts more osmotic pressure than glycogen. Why do insoluble particles, sodium chloride, exert more osmotic pressure than the non-ionizable? Ionizable particles more than the ionizable particles. For example, glucose. Ionizable particles ionize and thus have more osmotically active particles. Ionizable particles ionize and thus have more osmotically active particles. So, osmotic pressure of ionizable particles is more than the non-osmotic particles, glucose. One molecule of sodium chloride ionized into particles is more than the non-ionizable particles, glucose. One molecule of sodium chloride ionized into two particles. So, non-molecular of sodium chloride will exert double osmotic pressure than that of glucose. Endocytosis. Endocytosis definition. Endocytosis is defined as the transport mechanism by which the macromolecules enter the cell. Macromolecules cannot pass through the cell membrane either by active or by passive transport mechanism. Such substrates are transported into the cell by endocytosis. Types. There are various types of endocytosis based on the size of the particles being ingested in the regulatory requirements. Pinocytosis, ingestion of substrate that are in the solution of non-visible under microscope is called pinocytosis, for example, ingestion of protein, vitamin, etc. Phagocytosis, ingestion of substance and invisible under microscope such as bacteria, dead cell, tissue debris, etc. is called phagocytosis. Receptor mediated endocytosis allows uptake of specific molecules and occurs in specialized areas of membrane called Clathrin coated pits. Uptake LDL cholesterol. Catherine coated pits. Covalent development uptake. Non-Catherine. Non-concovalent endocytosis. Cell endocytosis. Endocytosis allows entry of molecules into the cell and cannot pass through the cell membrane either by active or by passive transport mechanism. Endocytosis is vital for survival, immunity, signaling, and therapy. Clinical and therapeutic relevance. Drug delivery. Nanoparticles exploit endocytosis for targeted therapy. Disease. Cystic fibrosis, defective CFTR endocytosis. Neurodegeneration, impaired synaptic vesicle recycling, for example, Parkinson's. Cancer, overactive growth factor receptors invade endocytotic degradation. Lung cancer. Exocytosis, the process by which cells release micromolecules in the exterior is called exocytosis. In this process, the substances are excluded from cell without passing through the cell membrane. This is reverse of endocytosis. Mechanisms of exocytosis. Substance to be released out of the cells inside the cell package secretory vesicles. Secretory vesicles. Vesicles fuse with the plasma membrane. Then plasma are secreted and vesicle membrane is incorporated in the plasma membrane. Role of calcium exocytosis. Calcium plays an important role during the release of some secretory substances such as neurotransmitters. The calcium ions enter into the cell and cause exocytosis. Importance of exocytosis. Exocytosis is involved in the release of secretory substances for cells. For example, neurotransmitters, hormones, enzymes, proteins are released from the cell by this process. Pathways. There are two pathways: constitutive pathway and inconstitutive pathway. Then there are ion channels. And then there are active transport and coactive transport and their differences, and a chart, and then membrane potential and action potential. Site of function. Sodium potassium pump significance, chemical importance, relationship difference with the secondary active transport functions. Physiological functions, chemical function, relation with the secondary active transport. Sodium potassium pump produces an inward directed sodium gradient which acts as energy for secondary active transport. Relation with the secondary active transport. Sodium potassium pump produces inward and direct sodium gradient which acts as energy for secondary active transport. Difference between sodium potassium pump and secondary active transport. Features. Sodium potassium pump active primary active transport, secondary active transport. Energy source. Directly uses ATP hydrolysis. Uses pre-existing ion gradient created by primary. Uses pre-existing ion. Directly uses ATP synthesis using pre-existing iron gradient created by primary. Pumps 3 sodium out and 2 potassium in ATP. Couples downhill movement of one solute to uphill movement on another. Rolling gradient creates ion gradient, high extracellular sodium and high intracellular potassium. Exploit gradients does not create them. Exploit gradients. Exploit P-type ATPase. Syn-porters and antiporters. Energetic, yes. Not net charge contributes to memory position. Net charge movement approximation depends. SIM ports SGLT, electronic and antiports, SGLT. Examples: sodium potassium ATPase, all cells, and hydrogen ATPase, lysosomes, hydrogen ATPase. SIM port sodium glucose transport SGLT, and antiport sodium calcium exchanger. Examples: biological role: maintains resting potential, cell volume and osmotic balance, absorbs nutrients, glucose, amino acids, and removes waste. Classify the membrane transport. On the definition: reading the membrane transport process. Sick cell syndrome, called cell volume dysregulation syndrome, refers to a pathogenic condition where cells lose their ability to regulate volume. Chronic cell shrinkage or swelling, where the cells lose their ability or swelling due to the impaired ion transmission mechanism, particularly ATP synthesis. Energy deprivation, ischemia, mitotic dysfunction, toxic exposure, IUBN or digotina, IUBN, alcohol, heavy metals, chronic kidney disease and diabetes. Clinical implications: brain cell and muscle cells, neurodegenerative disorders, Alzheimer's, where sodium potassium pump actively declines, Alzheimer's. Key symptoms and effects: cell shrinkage and cell swelling. Sodium accumulates inside, potassium leaks out. Accumulates inside, cell swelling and cell shrinking. Water influx due to osmotic imbalance. Organelles dysfunction, mitochondria swelling, necrosis, cell rupture. Cell shrinkage, sodium accumulates inside, potassium leaks out, cell death. Clinical implications: brain cell, neuronal shrinkage, conjunctive decline, muscular cells, impaired sodium potassium pump, kidney cells, dysregulation volume, hypertension, edema. The term cell sickness syndrome, it is not formally recognized medical or sacrificial diagnosis, but is osmotically used in broader context to describe the state of chronic cellular dysfunction caused by accumulating stress. The term cell sickness syndrome is not formally recognized by medical or scientific diagnosis, occasionally used in broader context to describe chronic cellular dysfunction caused by cumulative stress. What is cell sickness syndrome? Implies generalized cellular dysfunction. Cells struggle to maintain homeostasis due to oxidative mitochondrial dysfunction, toxic accumulation, chronic inflammation. Proposed symptoms: fatigue, muscle weakness, accelerated aging, and increased susceptibility to disease. How cell sickness syndrome (CSS) differs from cell sickness syndrome. How? Sick cell syndrome, cell sickness syndrome, and sick cell syndrome. Definition: informed term for global cell stress. Definition: informal term for global cell stress. Formal condition for sodium potassium pump failure. Cell sickness syndrome, sick cell syndrome, cell sickness syndrome. Informal term for global sex stress and formal term for sodium potassium pump failure. Vague, hostilic, specific, lack prior reviewed, well documented. Detox, antioxidants and lifestyle changes. Correcting the transport chain, ATP support. Discuss the process of glucose movement across the cell membrane with diagram. Process of glucose movement across the cell membrane. Sodium glucose cotransport. It occurs in the brush border in intestine, epithelium, and luminal part of the renal tubule cell membrane through sodium dependent glucose transport. Facilitated diffusion. This occurs in the basolateral part of the interstitial epithelium, renal tubule. Facilitated diffusion occurs in the basolateral part of the intestinal epithelium and renal tubule. Membrane through glucose transport. Through glucose transport. Renal tubule glucose transport. Insulin in muscles and most cells of the body and few exceptions. Diffusion. Diffusion may be defined as the movement of solute particles by the normal kinetic form of a higher concentration to a lower concentration. Example, exchange of O2 and CO2 in lung and tissue occur by diffusion. Type 2 type. Simple diffusion does not require carrier protein. Simple diffusion. Facilitated diffusion requires carrier protein and facilitated diffusion of glucose. Diffusion does not require carrier protein O2 CO2 and requires facilitated glucose. Factors controlling rate of diffusion: concentration difference, cross section area, distance between two regions, temperature, size of the particles, molecular weight, thickness of the cell membrane, lipid solubility, number of protein channels in the cell membrane. Importance of diffusion: exchange of O2 and CO2 in lungs and these tissues. Diffuse admixture of foodstuff and digestive juice occurs by diffusion. Absorption of protease, minerals, water soluble vitamins, etc. from GIT occurs in diffusion. Secretion of waste products into renal tubules occurs by diffusion. Diffusion sodium potassium through sodium potassium leak channels create resulting membrane potential. Exchange in capillary bed occurs in diffusion. Pneumonia, thickness of the alveoli membrane, less diffusion of O2 causes hypoxia. Thickness of the alveolar membrane. Mechanism of diffusion through the cell membrane. Simple diffusion. Through the interstitial lipid bilayer, the diffusion absence is lipid soluble. Exchange of O2 and CO2 in lungs, tissue occurs by diffusion. Admixture of pentose, minerals, water soluble vitamins, GIT occurs by diffusion. Section of waste products, minerals and vitamins. Diffusion. Diffusion of sodium potassium through the sodium potassium leak channel creates resting membrane potential. Exchange of capillarity bed occurs in diffusion. Pneumonia, thickness of alveoli membrane. Diffusion of O2. Simple diffusion. Through the interstices of lipid bilayer, its diffuse substance in lipid soluble. For instance, the lipid is soluble of oxygen, nitrogen, carbon dioxide, and alcohol are high, so that all these can dissolve directly in the lipid bilayer and diffuse through the cell membrane. Though water is channels that penetrate all the way through the same all large and broad transport proteins. Facilitated diffusion. Facilitated diffusion requires interactions of a carrier protein. A carrier protein aids passage to a molecule or ions through the membrane by binding chemically with them and shutting them through the membrane in this form. Facilitated diffusion. It is a passive transport process where molecules move down their concentration gradient. It is a passive transport process where move down their concentration gradient across a cell membrane. It's a passive transport process with the help of carrier protein channels. Unlike simple diffusion, it requires membrane proteins to assist the movement of molecules that are too large or polar to access the lipid bilayer freely. That are persist the movements membrane proteins. Why it is called as lipid bilayer membranes that assist the movement of molecules that are too large or polar to cross the lipid bilayer freely. Why it is called facilitated diffusion? Facilitated because protein mediated molecules transport proteins pass through the membrane. Example, GUT transport facilitates glucose and entry into cells. No energy required, like concentration gradient. Happens by concentration gradient. Overcomes membrane barrier, enables transport of molecules that cannot diffuse freely. Examples: glucose transport, GULT4, insulin regulated in muscle fat cell, GULT4, ion channels, aquapores and potassium leak channels. Clinical relevance: diabetes, impaired GULT4 functions, insulin resistance, cystic fibrosis, defected CFR channels, thick mucus. State the Fick's law and diffusion. The magnitude of the diffused capacity from one region to another is directly proportional to the cross-section area across which diffusion takes place and the concentration gradient or chemical gradient, which is difference. The magnitude of the diffusing capacity of one region to another directly the magnitude of the diffusing capacity from one region to another is directly proportional to the cross-section area across the diffusion. The magnitude of the diffusion capacity from one region to another is directly proportional to the cross-section area. Osmosis, the diffusion of solvent molecules across a semi-permeable membrane from the region of lower solute concentration to a region of higher solute concentration. It is called osmosis. Conditions of osmosis: two soluble of different osmotic pressure, presence of a semi-permeable membrane between the two solutes. Importance of osmosis: physiological importance: absorption from GIT, fluid exchange in capillary bed, regulation of urine formation, reabsorption of CSF, regulation of water balance in the body, maintenance of blood pressure. Clinical: infusion of isotonic solution. Infusion of isotonic solution 0.9 sodium chloride and 5 glucose maintains volume. In cerebral edema, hypertonic solution is used when causes osmosis of water from brain, thus reduces edema. Hypertonic solution is used. Hypertonic solution thus causing edema. When plasma proteins when RBC are taken in a hypertonic solution, water enters into the RBC and osmosis. RBC swells and eventually burst. Hemolysis. Purgatives prevent water absorption by osmosis from GIT. Thus, they cause dilution of intestinal contents and helps in deacidification. Thus, they help absorption from osmosis, prevents water from absorption. Purgatives. Thus, they prevents water absorption from osmosis. Thus, they can dilute intestinal contents and help in deacidification. Osmotic pressure: the amount of pressure required to prevent the osmosis is called osmotic pressure. Factors regulating osmotic pressure: in an ideal solution, osmotic pressure P is related to the temperature and the volume in the same way as pressure gas. Here N equals to number of osmotically active particles, R equals to gas content. Number of osmotically active particles and R equals to gas content. Number of osmotically active particles and number of gas content. From the equation we can find the factors. Number of osmotically active particles. Osmotic pressure is directly proportional to the number of osmotically active particles. Temperature proportional, volume is inversely proportional. Effect of ionization: ionizable solutes have more PO because ionizable solutes dissociate with two or more ions. Each of these ions then exerts OP individually. Ions of solute: number of particles. Effect of aggregation: some substances, for example soap, a molecule aggregates in solution, thus decreases number of osmolarity active particles. Note 12. Colloid osmotic pressure. Colloidal osmotic pressure: the osmotic pressure that is exerted by the colloid is called colloidal osmotic pressure. The osmotic pressure that is exerted by the colloid is called colloidal osmotic pressure. Osmotic pressure that is exerted. Value of colloidal osmotic pressure: colloidal exerts low osmotic pressure. Plasma proteins exert osmotic pressure. Colloidal exerts low osmotic pressure. Plasma proteins exert osmotic pressure of about 28 to 32 millimeter mercury, average 30. Plasma proteins exert low osmotic pressure. Importance: the capillary bed plasma colloidal osmotic pressure causes osmosis of tissue fluid from testis space back to the blood vessels. It plays important role in regulation of glomerular filtration. Discuss the role of osmotic pressure in maintenance of fluid volume. Role of osmotic pressure in maintenance of fluid volume. Osmotic pressure is pressure force that regulates fluid balance across the cell membrane and blood vessel by driving water movement. It ensures proper fluid distribution between the intracellular and extracellular compartments, maintaining homeostasis. Osmotic pressure is the pressure required for prevent water movements across a semi-permeable membrane due to solute concentration difference. It is governed by osmolarity and tonicity. Osmotic pressure gradient, tonicity. Intracellular fluid balance, cell volume stability. Water leaves the cell, cell shrinkage. Water hyponatremia. Water enters the cell, cell swelling. Regulated by sodium potassium ATPase pump, maintains osmotic pressure and aquapores. Plasma volume. Albumin generates colloidal osmotic pressure, retaining water in capillaries. Albumin generates colloidal osmotic pressure. Plasma volume. Albumin generates colloidal osmotic pressure, retains water in capillaries. Low COP, nephrotic syndrome, edema, interstitial fluid, balanced by Starling forces. Renal water reabsorption. Kidney adjust water reabsorption based on the osmotic gradients. ADH. Increase aquaporin 2 channels in collecting ducts. Water retention. Countercurrent multiplier creates a medullary osmotic gradient for urine contraction. Which one will exert more osmotic pressure, colloidal or crystalloid? Because osmotic pressure directly proportional to the number, not the size. Which one will exert more osmotic pressure, glucose or glycogen? Glucose is crystalloid and glycose colloid. Glucose exerts more osmotic pressure. Why do ionizable particles exert more osmotic pressure than non-ionizable particles? Endocytosis. Endocytosis is defined as the transport mechanism by which the macromolecules enter the cell. Macromolecules cannot pass through the cell membrane either by active or by passive transport mechanism. Such substances are transported into the cell by endocytosis. Endocytosis types. There are various types of endocytosis based on size, particles, based on ingestion and regular requirements. Pinocytosis. Ingestion of substances that are insoluble and not visible under microscope. It is called pinocytosis. Ingestion of protein, vitamin, etc. Phagocytosis. Ingestion of substances visible under microscope such as bacteria, dead cell, tissue debris, etc. Phagocytosis. Bacteria, dead cell, tissue debris, etc. Receptor mediated endocytosis. Clathrin mediated endocytosis allows uptake of specific molecules and occurs at specialized areas of membrane called clathrin coated pits. Uptake of LDL cholesterol. Clathrin coated pits. Clathrin coated pits. Clathrin coated pits. Caveolar dependent uptake. Non-clathrin, non-caveol endocytosis. Importance of endocytosis. Endocytosis allows entry of macromolecules into the cell which cannot pass through the cell membrane either by active or by passive transport mechanism. Endocytosis is vital for survival, immunity, signaling, and therapy. Endocytosis allows entry of the macromolecules into the cell and cannot pass through the cell membrane either by active or passive transport mechanism. Survival, immunity, signaling, and therapy. Clinical and therapeutic relevance. Drug delivery. Nanoparticles exploit endocytosis for targeted therapy. Cystic fibrosis, neurodegeneration, cancer. Exocytosis. The process in which cells releases macromolecules in the exterior is called exocytosis. This is process in the substance are excluded from the cell without passing through the cell membrane. This is reverse of endocytosis. synthesized cell package of secretory substances to be released out of the cell are synthesized in the cell and packaged to secretory vesicles. Synthesized in the cell and packaged to secretory vesicles. Vesicles fuse with the plasma membrane. The substance are recreated and the vessel membrane is incorporated in the plasma membrane. The substances are secreted and the vessel membrane is incorporated in the vessel membrane. Incorporated in the vessel membrane. Incorporated. Role of calcium in excitosis. Calcium ion plays an important role during the release of some secretory substance such as neurotransmitters. The calcium ions enter the cell and cause exocytosis. Importance of exocytosis. Exocytosis is involved in the release of secretory substances from the cells. For example, neurotransmitters, hormones, enzymes, proteins are released from the cell by this process. Pathways. There are two pathways for exocytosis. Constitutive pathway transports proteins with little or no passing or storage, present in most cells and used to export extracellular matrix protein. Non-constitutive pathways. Ion channels. Ion channels are transmembrane protein channels that allow the selective transport of various ions across the membrane. Voltage gated channels, ligand gated channels, and mechanically gated channels. The ion channels which open and close in response to changes in the membrane potential are called voltage gated channels. For example, potassium, sodium, chlorine channels and calcium channels. Ligand gated channels. These ion channels which open and close in response to building a specific ligand to receptor are called ligand gated channels. Acetyl chlorine channels. The channels are open and close in response to binding with a specific ligand to the receptors. Response: close, open or close in response to binding of a specific ligand gated channels. A specific ligand to the receptor, a specific ligand to bind of a specific ligand. For example, acetyl choline. Membrane potential. Response to changes in membrane potential. Membrane potential. Mechanical gated channels. Stimuli, pressure and touch. Different ion channels: sodium, potassium, chlorine, calcium, acetylcholine channels. Clinical importance: sodium channel inhibitor is used in heart failure. Local anesthetics act as blocking sodium channels. Calcium channel blockers used as hypertension, myocardial infarction, angina pectoris, and cardiac arrhythmia. Myocardial infarction and hypertension. Cl channels stimulator is used as a sedative drug. Importance of leak channel voltage and gated channels are chemical gated channels. Leak channels function: always open, allows ions to leak across the membrane down their electrochemical gradient. Leak channels allows leak down their electrochemical gradient. Maintains resting membrane potential. Leak: potassium ion leaks to polarize at -70 millivolt. Channels: set baseline electric channels. Is balance of ion water flow. Example: sodium leak channels in neuron prevents excessive depolarization. Located gated channels function: open close at respond to channels in membrane potential. Importance: generate action potential. Propagate electrical channels. Enable rapid response. Neurotransmitter release tracked by calcium influx. Neurotransmitter released triggered by calcium influx. Cardiac condition via voltage gated calcium channels. Channels in neurons and muscles. Examples: sodium channels, potassium channels, calcium channels. Ligand: open or close to a specific molecule. Ligand bond. Importance: synaptic transmission, sensory receptor, sensory perception, and cellular communication. Examples: GABA receptor, biological neurons, muscle homeostasis. Reference: some properties of ion channels. Comparison of transporters in ion channels. Transporters and ion channels bind solute and undergo conformation changes, transferring the solute across the membrane. Involved in passive On active transporters. Transport is significantly slower than via channels. Ion channels from pores membranes involves only in passive transport. Transports are significantly faster than via transporters. Secretion. Secretion is the process of produce release in useful substances in a gland or cell. GIT, lungs, submucosal glands, and mammary glands. Traits: active transport and passive transport. Movement occurs, energy, carrier protein, membrane, saturated carrier, direction of movement, source of an electrochemical gradient. Give the difference between secondary active transport and facilitated transport. Movement occurs, energy, direction, movement. Active transport, passive transport. Traits and secondary active transport and facilitated diffusion. Coordinate bioreactor and operate bioenergy. Traits: active transport, facilitated diffusion. How does co-transport differ from facilitated diffusion? Co-transport, facilitated diffusion. Mention the difference between primary active to secondary active transport. Subdivisions now two types: primary active transport, secondary active transport. Difference. Subdivisions now two types: co-transport and subdivisions now primary active transport. Two types: co-transport and counter-transport. Number of substance transported: one or more. For example, hydrogen pump and sodium potassium pump must be more than one. One or more must be more than one. Against the electrochemical gradient and on substance along the electrochemical against the electrochemical gradient substance. One substance along the electrochemical gradient and another substance against the electrochemical gradient. ATP of other high energy phosphate. Electrical gradient produced by primary transport. ATP have another high energy phosphate. ATP or other high energy phosphate. It occurs via membrane proteins that directly couple with ATP hydrolysis to solute movements. It occurs when uphill transport is compared via protein is coupled with the passive downhill transport in an ion. Difference between simple diffusion and active transport. Traits: simple diffusion, active transport. Towards concentration electrical gradient, against concentration electrical gradient. No required energy may or may not be present, may be present. Cause can operate directionally, usually under rational, unidirectional. Can operate birationally, usually unidirectional. Depends on them, does not depend on them. Difference between simple and facilitated diffusion. Simple diffusion, facilitated diffusion. Definition: carrier saturation of carrier, diffusion capacity, substance that are transported. Example: carrier needed, not needed. Facilitated diffusion. Traits: osmosis and diffusion. Fuck you. Movement, molecular challenge must be present. Permeable solvent not necessary. Osmotic pressure. Osmotic and diffusion. Movements of solvent solutions occurs from lower to higher concentration must be present. Same permeable solvent not necessary if contains as long as osmotic pressure exists. Duration: it occurs at every moment. Number of solute particles as per volume temperature. Concentration graded molecular weight. Difference between osmosis and active transport. Active transport: movement of solute molecules, movement of solvent molecules. Energy required, non required. From lower to higher concentration solute, from higher to lower solvent. Permeable of solute and solvent, permeable to solvent. Not required, sodium glucose. Secondary active. Membrane potential and action potential. Basic principles of electricity. Separated electrical charges of opposite signs have the potential to do work if they are allowed to come together. This potential is called an electrical potential. It is also called potential difference because it is determined by the difference in the amount of charge between two points. Write down the basic physics of membrane potential. Basic physics of membrane potential. There is a concentration difference of ion across the selectively permeable membranes. Basic physics of membrane potential. There is a concentration difference of ion across the selectively permeable membranes. Due to this ion concentration difference, separated electrical charges of opposite signs have the potential to do work if they are allowed to come together. This potential is called electrical potential. Due to this ion concentration difference across the selective permeable membrane, due to this ion concentration difference, separated electrical charges of opposite signs have the potential to do work if they are allowed to come together. This potential is called electrical potential. As this electrical potential occurs across the membrane, it is called membrane potential. Electrical potential exists across the membrane of virtually all cells of the body. Some cells, such as nerve cells and muscle cells, generate rapidly changing electrochemical pulses at the membranes called action potential. Membrane potential. The electrical potential difference across the membrane is called membrane potential. Types of membrane potential. It is two types. Resting membrane potential, action potential. Define membrane potential. Membrane potential. The electrochemical potential difference across the membrane is called membrane potential. Types of membrane potential. It is of two types: resting membrane potential and action potential. Give the importance of membrane potential. Importance of membrane potential. The membrane potential voltage difference across the cell membrane is crucial for cellular communications, homeostasis, and physiological functions. Below are the key roles. Electrosignaling excitable cells, neuron, generate action potential nerve impulses for rapid communications. Controls neurotransmitters release. For example, synaptic transmission. Action potential and neurotransmitter release. Neurons. Action potential and neurotransmitter release. Muscles. Triggers muscle contraction via depolarization of skeletal cardiac muscles. Maintenance of cellular homeostasis. Ion balance. The sodium potassium pump maintains gradient, preventing cell swelling of or shrinking. Ion balance. The sodium potassium pump maintains pH regulation. Proton H pump rely on membrane potential to acidify organelles. pH regulation. Proton pump rely on membrane potential to acidify organelles. So electric single excitable cells, maintenance of cellular homeostasis, nutrient and waste transport. Secondary active transport. The sodium gradient created by membrane potential drives uptake of glucose, amino acids, and other nutrients. Kidney function. Ion channels and pumps in nephron regulate electrical balance. For example, potassium excretion. Sensory transduction, vision. Photoreceptors in the retina responds to light via change in membrane potential. In retina responds to light via change in membrane potential. Vision. Photoreceptors. Touch. Mechanosensitive ion channels open in response to pressure. Cell volume regulation, osmotic balance. Potassium and chloride fluxes controlled by membrane potential prevent excessive water influx and exflux. Energy production. The proton motive force drives ATP synthesis in mitochondria. Immune response. T cell, B cell. Membrane potential changes active immune cells during infection. Potassium and chloride controlled by membrane potential prevent excessive water influx and exflux. Energy production. Mitochondria. Proton motive force derives ATP synthesis in mitochondria. Immune response, T cell, B cell. Membrane potential changes active immune cells during infection. Immune cells. Key takeaway summaries. Membrane potential is essential for rapid signaling, nerve and muscles, nutrient absorption, intestine, kidneys, cellular survival, volume, pH, kidney energy balance, and energy balance. Disruption led to neurological, muscular, and metabolic disorders. Reference: Physiology and Neuroscience. Resting membrane potential, RMP. Definition: the constant electrical potential difference across the cell membrane with the inside negative relative to the outside of the cell at rest is called resting membrane potential, RMP, or the resting membrane potential, RMP, is the steady voltage difference across a cell membrane when the cell is not actively transmitting signals. The resting membrane potential, RMP, is the steady voltage difference across a cell membrane when the cell is not actively transmitting signals. It is a fundamental property of all living cells, particularly crucial, crucial, critical, permanently critical in neuron and muscle cells. Where presence, it is found in almost all cells. Cause of development, RMP results from separation of excess of positive charges outside the cell and negative charges inside the cell at rest. Definition: the constant electrical potential difference across the cell membrane with the inside negative relative to outside is at rest. Nerve fiber minus 90, neuron minus 70, as a nodal fiber minus 55 to minus 60, cardiac ventral muscle minus 85 to 90, minus 90, smooth muscle minus 50 to minus 60, skeletal muscle minus 80 to minus 90, RBC minus 9. RMP of skeletal muscle of minus 80 MV and RMP of cardiac muscle of minus 80 MV. RMP of skeletal muscle minus 90 MV, RMP of cardiac muscle minus 80 MV. Importance of RMP: readiness of signaling, sets the baseline for action potential, rapid polarization to minus 40 MV. Ion homeostasis, prevents cell swelling by balancing osmotic forces. Nutrient transport, drives secondary active transfer, for example glucose uptake via SGLT. Why is RMP negative? RMP negative, the resting membrane potential is typically negative minus 70 MV in neurons due to several reasons. Selective permeability of the membrane. The membrane is more permeable to potassium ions than to sodium at rest because of leak channels. Sodium leak channels are more abundant than sodium leak channels. Potassium leak channels are more abundant than sodium leak channels. Movement of potassium increases a negative interior. As potassium leaves the channel, it makes positive charge with it, making the inside of the cell more negative relative to the outside. This creates an electrical gradient that opposes further potassium fluxation. Sodium potassium pump maintains the gradient. The pump actively transports 3 sodium out and 2 potassium in for each ATP consumed. This results in a net loss of positive charges from the cell, contributing to the negative RMP. Large ions, negatively charged protein and phosphate are trapped outside. Intracellular proteins, nucleic acids, and other ions cannot cross the membrane, adding to the negative charge inside. Summary: potassium efflux due to concentration gradient makes the inside cell negative. Summary: potassium efflux due to the concentration gradient makes the inside negative. Sodium potassium pump maintains ion gradient and contributes to negativity. Independent ions inside the cell add to negative charge. The RMP is a balance between potassium diffusion and sodium influence and active transport. Thus, the resting membrane potential is negatively permeable negatively primarily because of sodium potassium leaving the cell, while sodium enter the sodium potassium pump help fine tune the exact value. Reference Candle Swartz Principle of Neural Science. How is resting membrane potential produced and maintained in cell? Origin and maintenance of resting membrane potential RMP. The resting membrane potential RMP is produced and maintained by a combination of ion concentration gradients, selective membrane permeability, and active ion transfer mechanisms. Here's a detailed breakdown. Selective membrane permeability. Potassium leak channels dominated at rest. How is resting membrane potential produced and maintained in cell? The membrane is 100 times more permeable to potassium than sodium at rest due to potassium leak channels. Potassium effuses out down its concentration gradient, leaving behind negatively charged proteins and phosphates, which cannot cross the membrane. This creates a negative intracellular charge minus 70 microvolts in neurons. Equilibrium between diffusion and electrical forces. Nernst equation. The Nernst potential of potassium minus 94 millivolts is close to RMP because potassium is more permeable to ion rest. Potential 94 is close to RMP because potassium is the most permeable ion at rest. However, slight sodium permeability via leakage pulls RMP less negative values. However, slight sodium permeability via leak channels pulls the RMP to less negative values. Ion concentration gradients established by sodium potassium pump. Sodium potassium pump is actively transport 3 sodium out the cell and 2 potassium in the cell. Creates high extracellular sodium and high intracellular potassium 145 millivolts. The pump maintains these gradients, which are essential for generating resting membrane potential. Role of Goldman-Hodgkin-Katz equation. The action RMP is determined by the relative permeabilities of sodium, potassium, and chlorine. Since permeability of sodium is much more important than permeability of potassium as well as sodium, pK is greater than PNA. Potassium has the dominant effect. Role of the non-digestible ions, intracellular proteins, and organic phosphates. Large negatively charged molecules, for example, proteins, ATP, nucleic acids, are trapped inside the cell. They contribute to the negative resting potential by balancing the potassium efflux. Maintenance of RMP by the sodium potassium pump. Electrogenic effect prevents the ion equilibrium. Without the pump, sodium and potassium gradients would dispute over time, causing depolarization. The pump counteracts passive leaks, maintaining steady state resting membrane potential. The electrogenic effect -4 microvolt contribution helps maintaining the negative RMP. Resting membrane potential -86 plus -4 microvolt equal to -90 microvolt. Summary of key mechanisms: potassium efflux, sodium influx, potassium sodium pump trapped ions and JHK equation predicts RMP based on the ion permeabilities and concentrations. Makes insight more negative, slightly reduces negativity, maintains gradient and contributes -4 microvolt, add to negative charge inside. Conclusion: the RMP is primarily generated by sodium efflux but is slightly offset by potassium sodium efflux. The sodium potassium pump ATVS pump maintains the ion gradients while trapped ions stabilize the negative charge. Without these mechanisms, the cell would depolarize and lose excitability. Reference figure: establishment of resting membrane potential in nerve fibers. Under three conditions: when the membrane potential is caused entirely by potassium diffusion alone, when the membrane potential is caused by diffusion of both sodium and potassium ions, when the membrane potential is caused by diffusion of both sodium and potassium ions plus pumping of both of these ions by sodium potassium pump. Nernst equation: E equal to RT ZF R ln ion out by ion in. Goldman-Holkin-Katz equation. Goldman-Holkin-Katz equation: the membrane voltage depends on the conductance of ions and its concentration inside and outside. Conductance is the same as permeability for a changed ion. Generally, if it's going to move, it has to move through an open potential channel. Abbreviations: voltage across the cell membrane, outside voltage 0 because of the outside of grounded and so no charge can accumulate in the extracellular fluid. G ions: conductance of an ion, how easy it is for the membrane in equilibrium potential given by the Nernst equation of an ion. Name the factors responsible for generations of resting membrane potential. Factors responsible for generation resting membrane potential. Resting membrane potential RMP is generated by several key factors. Unequal ion distribution: high potassium inside, low potassium outside. High sodium outside, low sodium inside. Maintenance of sodium potassium pump. Selective membrane permeability: membrane is more permeable to sodium, less permeable to potassium, less permeable to sodium. Sodium efflux: main factor. The potassium moves out of the cell down its concentration gradient and leaves behind negative charges. Potassium phosphate inside. Sodium influx: minor influence. Small sodium leak into the cell makes RMP slightly less than potassium equilibrium potential -86 microvolt instead of -94 microvolt. Sodium potassium pump: sodium 3 sodium and out and 2 potassium in, contains -4 microvolt to RMP. Trapped ions: non-diffusible negatives. Large intracellular ions, proteins, organic phosphate cannot leave, adding to negativity. Summary: RMP is mainly due to selective permeable membrability, potassium efflux, sodium influx, sodium potassium pump, and trapped negative molecular inside. Action potential: the action potential AP is rapid temporary change in membrane potential caused by sequential ion effluxes, mainly sodium and potassium, and allows for single transmission. It begins with a sudden change from the normal resting negative membrane potential to a positive potential and then ends up back to the negative potential. Name the factor responsible for generation of resting membrane potential. Factor responsible for generation of resting membrane potential. The resting membrane potential RMP is generated by several key factors: unequal ion distribution, selective membrane permeability. Action potential definition: the action potential AP is a rapid temporary change in the membrane potential caused by the sequential ion fluxes that allows the single transmission. It begins with a sudden change from the normal resting negative membrane potential to a positive potential and then ends rapidly back to the negative potential. Stages of action potential. The successive stages of action potential are as follows. Resting stage. The constant electrical potential difference across the cell membrane with the inside negative relative to the outside of the cell at resting is called resting membrane potential. Dominant ion K. High permeable via leak channels. Sodium potassium pump maintains gradients. Trapped ions keep the inside negative. Depolarized stages. Rising phase minus 70 microvolt and plus 30 microvolt. The rapid rise of membrane potential in the positive direction due to rapid influx of sodium through voltage gated sodium channels is called depolarization. In large nerve fibers, the membrane potential actually overshoots beyond the zero level. Zero potential isopotential becomes somewhat positive. Triggered threshold minus 55 volt reached voltage gated sodium channels. Ion flux sodium rushes in, causes rapid unswing. Peak minus 30 millivolt. Repolarization stage, falling phase minus 30 microvolt to 70 minus 90 microvolt. Immediately after depolarization, the membrane potential reverse and fall rapidly towards the normal negative resting membrane potential due to rapid efflux of potassium through voltage gated potassium channels. This is called repolarization. Sodium channels inactivate cause after auto closing after 1 millisecond. Voltage gated sodium channels open. Delayed response. Ion flux. Potassium rushes out down its gradient. Restore negative membrane potential. Hyperpolarization positive after potential below minus 70 microvolt. After reaching the previous resting level, the membrane potential overshoots slightly in the hyperpolarized direction. It becomes more negative than its original resting level. This is called after hyperpolarization or positive after potential. Sodium channels remain open briefly. Overshoot R and P. Excess potassium channels remain open briefly. Excess potassium efflux makes membrane more negative, minus 80 millivolt. Eventually, potassium channels close and sodium potassium pump restores ion balance. Mention the ionic balance. Ionic fluxes of different phases of action potential. Ionic bases of ion flux of action potential. Phases, key ion movements and channel activity. Phase, resting, depolarization, repolarization, hyperpolarization. Key ion movements. Potassium leak, sodium influx, potassium efflux and excess potassium efflux. Nice to know after depolarization. When repolarization is about 70% completed, the rate of repolarization decreases and then the membrane potential approaches the resting membrane potential slowly. The slow fall of repolarization at the end of the process is called after depolarization. When the depolarization is about 70% complete, after depolarization, the rate of repolarization decreases. Membrane potential approaches resting membrane slowly. What is spike potential? Spike potential. The spike potential refers to a sharp, rapid rise of repolarized fall of the membrane voltage during action potential in excitable cells. It is the main upward and downward deflection in action potential graph. The key features of a spike potential: rapid depolarization, peak, overshoot, upshoot, overshoot, rapid repolarization, downshoot, and brief duration. Typically lasts about 1 to 2 milliseconds in neurons. What is called a spike? The spike, sharp needle-like and electrophysiological recording assemble a spike. Physiological Significance: neural communication. Spike potential allows rapid signal transmission along axons. All or no principle. Once the threshold is reached, the spike potential fires at maximum amplitude. Once the threshold is reached, the spike potential fires at maximum amplitude. Receptor potential. Define receptor potential. How can it be action potential generated from A? The receptor potential, also called generation potential, is a graded localized change in membrane potential produced in a sensory receptor, touch, light, sound receptors, in increased in stimulus. Generation of action potential from receptor potential to action potential, step by step mechanism. A stimulus active receptor, sensory receptor, IG, for example, touch, light, sound, intake stimuli. Stimuli opens mechanically, chemically, terminally graded ion channels, causing ion flow. Receptor potentials, gradiated potentials, local depolarized. For example, sodium influx generates a receptor potential. Amplitude varies with stimulus strength, decays passively out over distance. Transduction of generator potential. In a non-specialized receptor, receptors potentially directly triggers AP. In specialized receptors, receptors potential modulates neurotransmitter release into sensory neuron. Threshold reached. If depolarization at the trigger zone, for example, axon hillock, reaches threshold -54 microvolt, voltage gated sodium channels open. Rapid action potential. Action potential propagation. AP travels along the axon to the CNS, sensory neurons, and affect motor neurons. Example: Pacinian corpuscle, pressure receptor. Pressure deforms the receptor, opens sodium channels. Receptor potential. Generator potential in sensory neuron. If strong enough, AP is a sensory axon. What is all or none law? When a stimulus is above the threshold intensity, action potential is produced. Further increase in the intensity of the stimulus will produce no increment or other changes in the action potential. It occurs with the constant amplitude of other from, regardless the strength of the stimulus. If the stimulus is at or above threshold intensity, but the action potential fails to occur in the stimulus to establish the magnitude, action potential is therefore all or none in character and said to be obey all or none law. Importance of sodium potassium pump in resting membrane potential. The sodium potassium pump is critical for establishing the maintenance of resting membrane potential RMP neurons in muscle cells. The pump creates ion gradient needed for RMP. It has different interagenic effects. It supports potassium dependent RMP by maintaining high intracellular potassium. Blocking of action potential. Action potential can be blocked by disruption of ion flow, membrane excitability, or ion conduction. Hence are the primary methods. Sodium channel blockers. Example: tetrodotoxin and lidocaine. Blind to voltage gated sodium channels, prevent sodium influx, halts depolarization, complete AP blockade at sufficient doses. Hyperpolarization, increase sodium efflux and chloride influx. Demyelination. Physical method: nerve compression and cold temperature. Electrolyte imbalance: hyperkalemia, high potassium, hyperkalemia, and hypocalcemia, low potassium, high sodium, and low potassium. Legal examples: anesthesia, lidocaine blocks pain signals. TTX poisoning, paralysis due to neuromuscular AP blockade. Multiple sclerosis, AP failure cause muscle weakness and numbness. List of physiological difference between nerve and muscle excitable tissue. Physiological difference between nerve and muscle excitable tissue. Traits, nerves, muscles. Functional alignment, neuron, actin, myosin. Function, integration and transmission of nerve impulse, contraction and reflection. Resting membrane potential -70, resting membrane potential -90 microvolt, -70 microvolt. Stimuli for production of action potential, maybe electrical, chemical mechanism, maybe electrical, chemical, or medical. Plateau in action potential, plateau, absent, present. Contractile mechanism, absent, present. Secretory mechanism, present, absent. Neurosecretion and neurotransmitters. Neurons secrete neurotransmitters. Difference between the action potential of nerve fibers and cardiac muscle. Difference between the action potential and nerve fibers, cardiac muscle. Difference between action potential or nerve fiber, cardiac muscles. Difference between action potential and nerve fiber, cardiac muscle. Duration short, 1 to 4 ms. Cardiac muscle long. Phases: depolarization, repolarization. Cardiac muscle depolarization, plateau phase, and repolarization. Ion channels, ion involved, sodium influx, potassium efflux. Depolarization, repolarization. Sodium influx, potassium influx, and sodium influx, calcium influx, and potassium efflux. Plateau phase, absent, present. Refractory period, short and very long. All or none law strictly follows. Follows, but duration varies with heart rate. Function: rapid singular transmission, sustained contraction for pumping blood. Example cells: neurons, cardiomyocytes. Write down the difference between graded potential and action potential. Difference between graded potential, action potential. Graded potential, action potential. Graded varies with size of the initiating event, independent of the size of the initiating event. Cannot be summoned, can be summoned. Summation. Graded potential, action potential. Threshold, absent, present. Action, graded potential, action potential. Refractory period, absent, present. Refractory period. All in one law does not follow. It is conductant decrementally. It is conducted without decrement. Varies with initiating conditions, constant for given cell types. Ligand gated channels, voltage gated channels. Ligand gated channels are graded and voltage gated channels are action potential. Can be depolarized or hyperpolarized, only can be depolarized. Can be depolarized or hyperpolarized, can only be depolarized. Initiated by environmental stimulus receptors, by neurotransmitters, or spontaneously. Graded potential. Difference between receptor potential and action potential. Receptor potential: when a receptor is adequately stimulated or a non-propagated depolarized potential is developed, called receptor potential or graded potential. Action potential: rapid change in the membrane potential followed by a return of resting membrane potential is called action potential. It is non-propagated and graded. It is propagated and non-graded. It does not allow one or none law. Receptor potential: it allows one or none law. Follows. It has refractory period. It has not refractory period. It has refractory period. No refractory period. Increase in the intensity of the stimulus increased by multitude of RP. Increase in the intensity of stimulus, increase in frequent AP. Production of RP does not depend on AP. When RP reaches the threshold level, AP is produced. Action potential, rest moment potential. How is the action potential propagated along the nerve fiber? Propagation of action potential along a nerve fiber. In unmyelinated nerve fiber, continuous production of nerve. How action potential propagated along the nerve fiber. Propagation of action potential along the nerve fiber. In unmyelinated nerve fiber, continuous conduction by local currents. In myelinated nerve fiber, by saltatory conduction. Continuous conduction unmyelinated fibers. When an AP is generally in an axon helix depolarized open voltage gated sodium channels allowing the sodium influx. This depolarization spreads successively to adjacent regions due to local current flow between depolarized and resting membrane areas. When adjacent membrane reaches threshold, a new AP is generated, ensuring unidirectional propagation due to the refractory period. Saltatory conduction. Myelin acts as an insulin, preventing the flow of across the membrane at intranodes. AP jumps from one node to the Ranvier. AP action potential jumps to the one node to the next because depolarization occurs only at those exposed regions. High density of sodium channels. Increase conduction velocity and conserve energy. Nice to know key factors affecting propagation speed. Myelination via speed, saltatory conduction, axon diameter, large diameter, less resistance, and temperature. High temperature first ion channel kinase. Hand plate potential. The depolarization potential produced in the motor end plate in response to nerve impulse is called end plate potential. Mechanism of production. Arrival of nerve impulse at the motor nerve terminal. Release of acetylcholine from perisynaptic nerve vesicles. Binding of acetylcholine to end plate receptors. Opening of acetylcholine channels. Increase in sodium potassium conductance, sodium influx, end plate potential created. Importance of plate and end plate potential. When EPP reaches a critical magnitude, muscle action potential is produced, which leads to the muscle contraction. Glossary of the term describing membrane potential. Glossary, nice to know. Potential or potential difference. Potential or potential difference. Voltage difference between 2 points. Membrane potential or transmembrane potential. Voltage difference between the inside and outside of the cell. Resting membrane potential or resting potential. The steady transmembrane potential of cell that is not producing an electric signal. Graded potential. A potential channel change of variable amplitude and duration that is considered discriminantly. It has no threshold or refractory period. Action potential. A brief all or none depolarization of the membrane, reversing polarity in neurons. Synaptic potential, receptor potential, pacemaker potential are spontaneous occurring graded potential change that occurs in certain specialized cells. Threshold potential, the membrane potential in which action potential is initiated. Muscle physiology. Physiology, chapter 5. Describe the structural and functional components of skeletal muscle. Structural and functional components of skeletal muscle. Organization. Skeletal muscle is made out of individual muscle fibers that are the binding blocks of the muscular system. Each muscle fiber is a single cell that is multinucleated, long, cylindric, and surrounded by a cell membrane, the sarcolemma. The muscle fibers are made out of myofibrils, which are invisible into individual filaments. The neck filaments are made out of myosin, and the thick filaments are made out of myosin, and the thin filaments are made out of actin, tropomyosin, and troponin. Striations. The myofibrils have alternate light and dark bonds. These bonds give the muscle their strained appearance. The light bond bands contain only actin filaments and are called I bands because they are isotropic to polarized light. The light I bond is divided by the dark Z line where the end of the actin filaments are attached. The dark bonds contain the myosin filament as well as the end of the actin filaments where they overlap the myosin and are called A bands because they are anisotropic to polarized light. The lighter H bands in the center of the A bands are the regions where when the muscle is relaxed the thin filament do not overlap the thick filament. A transverse M line is seen in the middle of the H band. The area between the two adjacent Z line is called sarcomere. Sarcotubular system. It consists of T tubules and sarcoplasmic reticulum SR. The SR with its terminal cisterns surrounds each myofibril. The T tubules are originated from the sarcolemma and contact the myofibril between the A and the I bands. The function of T tubules is the rapid transmission of the action potential from the cell membrane to the fibril in the muscle. The SR stores calcium in high concentration which is released into the sarcoplasm when action potential appears in the adjacent T tubules. Short note: sarcomere definition. Short note: sarcomere definition. Sarcomere is defined as the structural and functional unit of the skeletal muscle. It is also called the basic contractile unit of the muscle. The area between the two adjacent Z lines in a myofibril is called a sarcomere. Extent: each sarcomere extends between two Z lines of myofibril. Each myofibril contains many sarcomeres arranged in series throughout its length. Average length of each sarcomere in relaxed muscle is 2 to 3 micrometer. Sarcomere definition, extent, components. Electron microscope study of sarcomere. The sarcomere consists of many thread-like structures called myofilaments. Myofilaments are of two types: actin filament and myosin filament. Figure: sarcomere is resting muscle. Sarcomere is resting muscle. Contracted muscle during contraction. A. Contracted muscle during contraction. B. Contracted muscle B during contraction. Z line comes close. H and I bonds are reduced and no charge in A band. Name the contractile units of muscle. Contractile units of muscle: thick filaments which are made out of myosin, thin filaments which are made out of actin, tropomyosin, and troponin. Myosin molecule. The myosin molecule is composed of 6 polypeptide chains, 2 heavy chains, and 4 light chains. Part of myosin molecule. Each myosin molecule has 2 parts. Tail portion: it is made out of 2 heavy chains which twist around each other in front of a double helix. Tail portion. are head portion. It is made out of 2 light chains and part of 2 heavy chains. Head portion. Binding sites of myosin head. 2 binding sites. One side for actin filament and other side for ATP molecule. Actin molecule. The backbone of the actin filament is a double-stranded F-actin protein molecule. Each actin molecule is the polymer of a small protein known as G-actin. Each F-actin molecule has an active site to which the myosin head is attached. Tropomyosin. About 40 to 60 tropomyosin molecules are situated along the double helix strand of actin filament. In relaxed condition of the muscle, the tropomyosin molecules cover all the active sites of actin molecules. About 40 to 60 tropomyosin molecules. Contraction occurs only when an appropriate single causes a conformation change in the tropomyosin that uncovers active site on the active molecule and initiates contraction. Troponin. These protein molecules are actually complexes of 3 loosely bound protein subunits, each of which plays a specific role in controlling muscle contraction. It is formed by 3 subunits: troponin 1, which is attached to F-myosin, troponin I, F-actin, troponin T, which is attached to tropomyosin, and troponin C, which is attached to calcium ions. I is F-actin, tropomyosin is T, C is calcium ions. The strong affinity of the troponin for calcium ion is believed to initiate the contraction process. What are the steps in muscle contraction and relaxation? Steps in skeletal muscle contraction: discharge of motor neuron, release of neurotransmitters, acetylcholine at motor end plate, discharge of motor neuron, skeletal muscle, motor neurons, neurotransmitters, acetylcholine, binding of acetylcholine to nicotine acetylcholine receptor, in increased sodium and potassium conductance in end plate membrane. Generation of end plate potential, generation of action potential in muscle fibers, inward spread of depolarization along T tubules, release of calcium from terminal cisternae of sarcoplasmic reticulum, and diffusion of thick and thin filaments, binding of calcium to troponin C, uncovering myosin bind sites to actin, formation of cross linkage between actin and myosin, and sliding of thick and thin filaments producing movement. Steps in relaxation: calcium pumped back into sarcoplasmic reticulum, release of calcium 2 plus from troponin, cessation of interaction between actin and myosin. What is power stroke? Power stroke definition: the power stroke is the pivotal movement of the myosin head that generates force and pulls the actin filament towards the center of the sarcomere, leading to muscle contraction. It occurs when myosin released ADP and PI, causing a conformational change that strokes the actin filament. Molecular mechanism of a skeletal muscle contraction or sequence of events in a power stroke: excitation, contraction, coupling, motor neuron action potential, acetylcholine released in an MJ, muscle fiber depolarization, neuromuscular junction, depolarization spread of via T tubules, calcium released in sarcoplasm, cross bridging of acycling, sliding filament mechanism. Calcium binding to troponin. Calcium binds to troponin C, shifting tropomyosin away from actin's binding site. Exposes myosin binding sites on actin. Myosin binding to actin, cross bridge formation. Myosin head pre-energized with ADP hydrolysis. ADP plus PI phosphate binds to actin. Power stroke. Release of PI and then ADP causes myosin head to pivot, pulling actin towards amylin. ATP binding to detachment. New ATP binds to myosin. Reduce actin affinity. Myosin detaches. Recovery stroke. Re-energization. ATP hydrolysis to ADP plus PI. Re-coking of the myosin head for next cycle. Relaxation. Calcium pumped back into smooth reticulum via SCRCA pump. Tropomyosin reblocks actin sites. Muscle relaxes. Textbook reference. Briefly write the walk-along theory of muscle contraction. Mention the role of ATP on it. Walk-along theory definition. The walk-along theory, also called the sliding filament theory, explains how muscle contraction occurs at the molecule level through the interaction of actin and myosin filaments. Walk-along theory definition. The walk-along theory, also called the sliding filament theory, explains how muscles contraction occurs at the molecular level through the contraction of actin and myosin filaments. Definition. The walk-along theory explains how myosin heads move along the actin filament in a cyclic, stepwise manner to produce muscle contraction. It is a refined version of the sliding filament theory, emphasizing the coordinated movement of multiple myosin heads for smooth and effective force generation. It is also called Rach theory or hand-over-hand model. Key steps of walk-along mechanism. Asynchronous myosin head movement. Not all myosin head binds and pulls simultaneously. Instead, they work in a staggered fashion to maintain continuous tension. While one myosin head is attached and pulling power stroke, another is detached and recocking. Sequential power strokes. After a myosin head completes its power stroke, ADP plus PI release, it binds a new ATP, detaches and re-energizes. Meanwhile, another myosin head binds further down the actin filament and executes its power stroke. Continuous filament movement. The alternating actin of multiple filament heads anchors the actin filament, slides smoothly towards the amylin, towards the jerky motions. Difference from conventional sliding move filament theory. The classic model assumes simultaneous pulling, but the walk-along theory explains how smooth contractions occurs due to sequential myosin head enlargement. Short note. Role of ATP in muscle contraction. Role of ATP muscle contraction. Energy liberated by breakdown of ATP is responsible for the following activities during muscle contraction: spread of actin action potential into the muscle, liberation of calcium ions from cisternae of L tubules into sarcoplasm, movement of myosin head, sliding mechanism. Steps of muscle contraction. Contractile proteins: myosin, actin, tropomyosin and troponin. Source of energy: ATP. Power stroke, the process by which the contraction of muscle is brought about is a sliding of a thin filament over the thick filament. The sliding during muscle contraction occurs when the myosin heads bind firmly to the actin, bend in the junction of the head of the neck, and then detach. This is called power stroke. Neuromuscular junction. Draw and label a neuromuscular junction. Nerve terminal, glial cell, active zone. Example how nerve impulse is conduced to skeletal muscle. Mechanism of neuromuscular transmission to skeletal muscle. Steps of neuromuscular transmission. Arrival of impulse, action potential at axon terminal of the motor neuron. Opening of voltage gated calcium channel. Entry of calcium ion to the axon terminal from the sympathetic cleft. Calcium causes fusion of acetylcholine vesicles with parasympathetic membrane. Release of acetylcholine into the sympathetic cleft by exocytosis. Acetylcholine diffuses through the synaptic cleft. Acetylcholine binds with nicotine type of acetylcholine receptor in the membrane of motor and plate. Opening of acetylcholine channels. Influx of sodium through the acetylcholine channel. Generation of end plate potential. When end plate potential becomes large enough, voltage gated sodium channels open. Sodium influx into the muscle fiber through sodium channels. Production of action of potential in the muscle. This is how impulse action potential is transmitted into the muscle. The muscle action potential in turn initiates muscle contraction. Structured essay questions. Neuromuscular junction. State the importance of neuromuscular junction and mention the steps of neuromuscular transmission, muscle contraction and relaxation. Neuromuscular junction. Components of neuromuscular junction: axon terminal, synaptic cleft, motor and plate. Steps of neuromuscular transmission. Sequence events of smooth muscle contraction and relaxation. Binding of acetylcholine to muscarine receptors. Increased influx of calcium plus into the cell. Activation of calmodulin dependent myosin light kinase chain. Phosphorylation of myosin. Increased myosin ATPase activity and binding of myosin actin. Contraction. Diphosphorylation of myosin by myosin light chain phosphatase. Relaxation or sustained contraction due to the latched bridge and other mechanisms. What is excitation contraction coupling? Excitation contraction coupling, the process by which depolarization of the muscle fiber initiates contraction, is called excitation contraction coupling. The process by which depolarization of muscle fiber initiates contraction is called excitation contraction coupling. Mechanism. Excitation of muscle. The excitation potentials in the skeletal muscle develop only when motor nerve impulses are transmitted into the skeletal muscle. Excitation muscle. The action potential in smooth muscle develop in response to excitatory neurotransmitters or hormone or from spontaneous oscillations of membrane voltage. Contraction of muscle. After contraction, intracellular calcium levels increase. Interaction between actin and myosin and the muscle contracts. Contraction of muscle. Excitation of muscle mechanism. In all types of muscle, the key event causing contractile is increased intracellular calcium concentration. Thus, calcium ions are the linking or coupling agent between those two different types of phenomena: excited electrical and contractile. Contraction mechanical. In short, development of action potential in the muscle cell. Excitation increased in intracellular calcium concentration. Calcium binds with troponin C in skeletal and cardiac muscle, or the calmodulin in smooth muscle. Muscle contraction. Action potential in TTBules, intracellular calcium 2 plus and muscle tension. Response time. Why calcium is important in skeletal muscle contraction. Role of calcium in muscle contraction. Calcium loss play a central role in initiating, sustaining and terminating muscle contraction. They act as the critical link between electrical and excitation, action potential and mechanical contraction, sliding of filaments. The key functions of sodium in muscle contraction: triggering contraction, excitation, contraction, coupling. Acting filaments in TTBules, calcium release from smooth reticulum, rapid sarcoplasm, calcium 2 plus from 0.1 microam to 1 to 10 microam, initiates contraction, triggering concentration, binding of troponin, unmasking actin sites. Calcium binds with troponin C on the thin filament, causes conformational change in the troponin-promyosin complex, exposed myosin binding sites on the actin, allows cross-bridge formation between the actin and myosin. Sustained contraction. High calcium maintains tropomyosin in open position, permits the repeated power strokes. Without calcium, tropomyosin blocks actin sites, preventing contraction. Termination of contraction. Calcium ATPase sarca pump actively pumps calcium back into smooth reticulum. Sarcoplasmic calcium fails. Calcium dissociates from tropomyosin. RE blocks actin muscle reflexes. Clinical and physiological importance. Hypocalcemia, low calcium, muscle weakness, tetany. What are the difference between skeletal and cardiac muscle? Difference between skeletal and cardiac muscle. Traits: skeletal muscle, cardiac muscle. Excitability: greater, lesser. Rhythmicity: absent, present. Fatigability: can be fatigued, cannot be fatigued, cardiac. Tetranization: possible, not possible. Plateau in action potential: absent, present. Plateau in action potential skeletal muscle: absent. In the cardiac muscle: present. Alternation of contractile response: little or marked, cardiac muscle. Mode of action: voluntary, involuntary. Source of calcium that initiates contraction: intracellular, extracellular. Anatomic properties: size, very long and uniform, little and very variable. Nucleus, multiple, flattened and lie peripherally. Cardiac, single, oval and lie centrally. Cause strength of Murphy: conspicuous, less conspicuous. Branched of fibrils: absent, present. Intercalated disc: absent, present. Syncytium does not act on syncytium, act on syncytium. Does not act on syncytium. Variations in number of active fibers: marked, little or none. Location: in association with bones in the heart. Write down the physiological properties of a skeletal muscle. Physiological properties of a skeletal muscle. Excitability. Excitability is defined as the reaction or response of a tissue of irritation or stimulation. It is a physiochemical change. Stimuli which can excite the tissue are of four types: mechanical stimulus, electrical stimulus, terminal stimulus and chemical stimulus. Pinching, mechanical stimulus, electrical stimulus, electric shock, terminal stimulus, applying heated glass rod or ice pieces, and chemical stimulus, applying chemical obstruction like acids. Contractibility. Contractibility is the response of the muscles to stimulus. Contraction is defined as the internal events of muscle with change in either length or tension of the muscle fibers. Types of contraction. Muscle contraction is classified into two types based on change in length of muscle fiber or tension of the muscle. Isotonic contraction. Here the tension remains the same and length of the muscle fiber is altered. Iso same tonic tension. Example, simple flexion of arm. Isotonic contraction. Isometric concentration. Here the length of the muscle fiber remains the same and the tension is increased. And the length of the muscle fiber remains the same but the tension is increased. Example, pulling any heavy object. Fatigue. Fatigue is defined as the decrease in muscular activity due to repeated stimuli. When stimuli are applied repeatedly after some time, the muscle does not show any response to stimulus. This condition is called fatigue. Refractory period. Refractory period is the period at which the muscle does not show any response to a stimulus. It is because already one action potential is in progress in the muscle during that period. The muscle is unexcitable to further stimulation until it is repolarized. Refractory period of two types: absolute refractory period and relative refractory period. Absolute refractory period. It is the period during which the muscle does not show any response at all, whatever may be the strength of the stimulus. Relative refractory period. It is the period during which the muscle shows some response if the strength of the stimulus is increased to maximum. Muscle tone. Muscle tone is defined as continuous and partial contraction of the muscle with certain degree of vigor or tension. Define isotonic and isometric contraction with example. Isotonic contractions. Muscle contraction is said to be isotonic when it does shorten with the tension of the muscle remaining constant. Isotonic contraction. Muscle contraction is said to be the isotonic when it does shorten with the tension on the muscle remaining constant. Example, this form of muscle work is carried out when walking or when lifting a load. Isometric contraction. Muscle contraction is said to be isometric when the muscle does not shorten during contraction. Isometric contraction. Example, such contractions occur in life when we simply maintain a posture against gravity. Since work and production of force times distance, W equal to F xd. Isotonic contraction do work, whereas isometric contraction do not. Skeletal muscle could be fatigued, but cardiac muscle never. Skeletal muscle and cardiac muscle have different physiological properties and functional demands, which explains why skeletal muscle can fatigue while cardiac muscles, the heart, do not. There are key reasons: energy supply and mitochondrial density. Skeletal muscle relies on glycolysis anaerobic during intense activity, leading to lactic acid building up in the fatigue. Cardiac muscle rich in mitochondria, 25 to 30% in cell volume, uses aerobic resorption, ensuring continuous ATP supply without fatigue. Fuel utilization. Skeletal muscle uses glycogen storage, which depletes quickly. Fuel utilization uses glycogen storage. Glycogen stores. Cardiac muscle prefers fatty acids, glucose, and lactic from blood, ensuring sustained energy. Calcium handling. Skeletal muscle as a released calcium rapidly but can deplete. Cardiac muscle uses calcium induced calcium release, CICR, with efficient CERC pumps, preventing calcium depletion. Oxygen dependence. Skeletal muscle can work anaerobically, ensuring pH droplets and fatigue. Calcium hardening. Skeletal muscle sodium smooth reticulum releases calcium rapidly but can deplete. Cardiac muscle uses calcium induced calcium released in CICR with efficient CERCA pumps, preventing calcium depletion. Oxygen dependence. Skeletal muscle can work anaerobically, causing pH drop, acidity, and fatigue. Cardiac muscle strictly aerobic, stops contracting if O2 is low, prevents fatigue but risk ischemia. Functional demand and rest period. Skeletal muscle fatigue contractions allow rest and recovery. Cardiac muscle contractions rhythmic activity, no rest. Evolved to resist fatigue for long function. Consolation. Cardiac muscle is high mitochondrial density, aerobic metabolism, efficient calcium handling, and constant blood supply, preventing fatigue, unlike skeletal muscle. The patch clamp method for recording ion current flow through simple protein channels is illustrated in figure 4.b. A microscope with a tip diameter of only 1 to 2 micrometers is abrupted against the outside of the cell membrane. Suction of this applied outside of the pipette to pull the membrane against the tip of the pipette, which creates a seal on the edges of the pipette, not cell membrane. The results minute membrane patch of the pipette touches which electrical current flow will be recorded. The small cell membrane patch at the end of the pipette can be torn away from the cell. The pipette with the sealed patch is then inserted into a free solution. Allows the concentration of both inside and outside of micropipette solution and clamped voltage below. Facilitated diffusion. Patch clamp method of recording current flow through the single channels. Membrane diffuse. And then facilitate affects rate of diffusion. Osmosis across a diffusion of water. Osmotic pressure. By far the most abundant substance that diffuses through the cell membrane is water. Enough water ordinarily diffuses in each direction through the blood vessel membrane per second is equal to 100 times the volume of cell itself. Yet the amount that normally diffuses in two directions is balanced so precisely that zero net volume of water currents. Therefore, the volume of the cell remains constant. However, under certain conditions, a concentration difference of water can develop across the membrane. When this concentration difference for water develops, net movement of water does occur across the cell membrane, causing the cell to swell or shrink. Depending on the direction of the water movement, this process of net movement of water caused by a concentration difference of water is called osmosis. To illustrate osmosis, let us assume the conditions shown in figure 4.10 with pure water on the side of the cell membrane and solution, sodium chloride on the other side. Water molecules pass through the cell membrane with ease, whereas sodium and chloride pass through with difficulty. Therefore, sodium chloride solution is usually a mixture of permanent water molecules. The non-permanent sodium are chloride ions, and the membrane is said to be selectively permeable water, but much less so to sodium chloride. Yet the presence of sodium and chloride has displaced some of the water molecules on the side of the membrane, whereas these ions are present and therefore has released the concentration water molecules are less than that of pure water. As a result, the example shown in figure 4.10, more water molecules strike the channels on the left side, while the pure water on the right side. Solution. Osmotic pressure. If the figure enumerate pressure were applied on the solution chloride, osmotic of water into the solution would be slowed, stopped, and even reversed. The amount of osmotic pressure required to stop osmosis is called osmotic pressure of sodium chloride solution. The principle of pressure difference opposing to osmosis is demonstrated in figure 4.11. Osmotic pressure difference. Osmotic of water from chamber B into chamber A causes the level of fluid columns to become further and further apart until eventually pressure difference develops between the two sides of the membrane and great enough to oppose the osmotic effect. The pressure difference causes the membrane at this point is equal to osmotic pressure of solution that contains the non-diffusible solute. Relationship of osmolarity and osmotic pressure at a normal body temperature, 75 concentration of osmoles per liter. Osmolarity is the osmolar concentration expressed as osmoles per liter of solution rather than osmoles per kilogram in water. Osmolarity is the osmolar concentration expressed as osmoles per liter of solution rather than osmoles per kilogram of water. Although strictly speaking, this is osmolar per kilogram of water is that determines the osmotic pressure. The quantitative differentiate between osmolarity and osmolarity are less than 1%, such as the body, because far more practical to measure osmolarity than osmolality. Measuring osmolarity is the actual practice in physical studies. Osmolarity to express the concentration of solution in terms of number of particles, a unit called osmol instead of grams. Resting membrane potential of neurons. The resting membrane potential of large nerve fibers when they are not transmitting nerve signals is about minus 70 millivolts. That is the potential inside the fiber of 70 millivolts or more negative than the potential of the intercellular fluid on the outside of the fiber. In the next few paragraphs, the transport properties of resting membrane potential for sodium and potassium and the factors determine the levels of the resting potential are explained. Active transport of sodium potassium ions through the membrane of sodium potassium pump. Recall from chapter 4, all the cell membrane potential of the body have powerful sodium potassium pump. Sodium potassium releases. There are sodium outside is 182 microequivalent per liter, 142 microequivalent per liter. Sodium inside 14 microequivalent per liter. Potassium outside 4 equivalent per liter and inside 140 equivalent per liter. Sodium inside, the ratio of these two prospective ions from inside to outside are follows. Inside outside ratio 1, potassium inside outside ratio is 3.5. Leakage of potassium through nerve cell membrane. The right side of the figure shows a channel protein, sometimes called a tendon pore domain potassium leak channel and nerve channels through the potassium ions. Even in resting basic structure of potassium channels are described in chapter 4. Origin of the normal resting membrane shows the important factors to establishment of normal resting potential. They are as follows. Contribution of potassium different potential. In figure 5.5a, we assume that the only movement of ion channels the membrane is diffusion of potassium ions as demonstrated by the open channels between the potassium symbol inside the sodium the membrane. Because the high ratio of potassium ions inside is about 3.35:1, the numbness potential corresponds to this ratio is minus 94 millivolts. Because the logarithm 94 is 1.54, this multiplied by minus 61 millivolts is 94 millivolts. Therefore, the potassium ions were the only factor causing the resting membrane potential. The resting potential inside the fiber would be equal to minus 94 millivolts as shown in the figure. Contribution of potassium diffusion potential. In the Figures 5.5a. We assume that only the movement of ions through the membrane is diffusion of potassium ions, as demonstrated by the open channels between the potassium symbol inside and outside the membrane. Because high ratio of potassium ions inside to outside 35:1 is the nullness potential corresponding to the ratio is minus 94 millivolts. Because of the logarithm of 35 is 1.54 and this multiplied by 61 millivolts is 94. Contribution of sodium diffusion through the nerve membrane shows the additional site of permeable nerve membrane. Sodium causes ions caused by the multiple diffusion sodium ions through sodium leak channels. The ratio of sodium ions from inside to outside of the membrane is 0.1, which gives a calculated earnest potential of the sodium shows in millivolts. How do these interact with each other? And that will be summarized potential. The question can be answered using Boltzmann's equation. Equation described previously. Can see the membrane potential slightly. Measurable sodium diffusion potential contributes to far more membrane potential diffuse of sodium of normal fiber. The permeability of sodium potassium is about 100 times as great as the permeability of sodium. 100 times as 100 times as part of 100 times as great as the permeability of sodium. Using this value of the Goldman equation, that considering only sodium and potassium gives potential inside the membrane of minus 86 millivolts, which is near the potassium potential shown in figure. Contribution of sodium potassium pump. The sodium potassium pump shows to provide an additional contribution to the resting potential. The figure shows that continuous pumping of these sodium ions to the sodium cores of each two potassium ions pumped to the inside of the membrane. The pumping of more sodium ions to the outside of the membrane than potassium ions being pumped in the inside causes continual loss of positive charges outside the membrane, creating an additional degree of negatively about minus 4 millivolts additional. Beyond it can be therefore, as shown in figure 5.5, the net membrane potential when all these factors are operative at the same time is about minus 90 millivolts. However, additional ions such as chloride must be considered calculate membrane potential. In the summary, diffusion potential alone caused by potassium or sodium diffusion gives a membrane potential about minus 6 millivolts, with almost all these being determined by potassium diffusion. An additional to minus 4 millivolts is the contribution to the membrane potential potassium. A additional minus 4 millivolts is then contributed to the membrane potential by continuously acting on electrogenic sodium potassium pump. By membrane potential, neuron action potential. Neuron cells are transmitted by action potential, which are rapid changes in the membrane potential that speed rapid along the nerve fibers. Each action potential begins with a sudden change from the normal resting negative membrane potential to positive potential and ends at the almost equal rapid change back to the negative potential. To conduct a nerve signal, the action potential moves along the nerve fiber until it comes to the fiber's end. The upper panel of figure 5.6 shows the changes that occur at the membrane during the action potential, with the transfer of positive charges into the interior of the fiber at the onset of return positive charges. The low panel shows graphically the successive changes in membrane potential over a few 10,000 of a second, illustrating the explosive onset of the action potential and almost equal rapid recovery. The successive stages of the action potential are as follows. Resting stage. The resting stage is the resting membrane potential and action potential begins. The membrane is used to be polarized during the stage because of the minus 70 millivolts negative membrane potential that is present. Depolarized state, repolarized state, voltage sodium channel, potassium channels, active sodium channel, inactive sodium channel. During this effective stage, sodium ions can pour inward through the channel, increasing the sodium permeability to the membrane as much as 500 to 5000 fold. Inactivation of sodium channel, voltage gated potassium channel and its activation. Full recovery of the resting membrane potential within another few 10,000 of a second. The voltage cam method of measuring the effective voltage of opening and closing channels. In the conductance of potassium ions is 50 to 100 times as great as the conductance of sodium ions. Initiation of the action potential. A positive feedback cycles open sodium channels. 60 millivolts towards the zero level. 60 millivolts towards the zero level. Raising voltage gated channels. Propagation of action potential. The positive charges of the voltage of a distance of 1 to 3 millimeters inside the large myelinated fiber above the threshold voltage, initiating an action potential. Therefore, the sodium channels in these new areas immediately open as shown on figure B, and the explosion action potential spreads. Initiation of action potential occurs only after the threshold potential is reached. In an action potential will not occur until the initial raise of membrane potential is great enough to create the positive feedback described in the preceding paragraph. It occurs when the number of sodium ions enter the fiber is greater than the number of potassium ions leaving the fiber. A sudden raise in the membrane potential of 50 to 30 millivolts usually required. Therefore, a sudden raise in the membrane potential of large nerve fibers 70 millivolts up to 55 millivolts, usually cause the explosive development of action potential. This level of minus 55 millivolts is said to be the threshold of stimulation. Direction of propagation, or on nothing principle. The establishment of sodium potassium ion gradients after action potential are compared importance of energy metabolism, relative in some action, rhythmicity of some exchange tissue and discharge. 1. Rhythmic beat of the heart, rhythmic peristalsis of the intestine, and neural events such as rhythmic control of breathing. Receptive self-improved discharge occur normally in the heart. Rhythmic of some excitable tissues, plateau in some action potentials. Impulses heart protrusion. In some cases, the excited membrane does not repolarize immediately. After depolarization, instead the potential remains on a plateau near the peak of the spike potential for many milliseconds before the depolarization begins. Such plateau is shown on figure. Plateau is greatly prolonged period of depolarization. The type of action potential occurs in the heart muscle fibers where the plateau lasts for as long as 0.2 to 0.3 milliseconds and causes contraction of the heart muscle lasts for the same long period. Usually voltage activated sodium channels called fast channels and voltage gated calcium sodium channels, which are shown on open and therefore are called channels. The re-excitation process necessary for a spontaneous rhythmicity. For a spontaneous rhythmicity to occur, the membrane, even in its natural state, must be permeable enough to sodium ions or calcium or sodium ions to show sodium calcium channels to allow automatic membrane depolarization. Some sodium, therefore, the following heart is only 60 to 70 millivolts, which is not enough to negative voltage to keep the sodium potassium channels totally closed. shows that the resting moment potential of rhythmic control center's heart is only 70 to 75 millivolts. Some sodium calcium channels flow inward, negatively cooled. Therefore, the following sequence occurs. Some sodium and calcium channels flow inward. This activity increases the membrane voltage positive direction, which follows the increased membrane permeability. Sodium and potassium flow inward. Still more ions flow inward. The permeability in each more and so on until an action potential is generated. Then the end of the action potential, the membrane is repolarized. After another delay of milliseconds or seconds, the spontaneous excitability causes depolarization again and a new action potential occurs spontaneously. This cycle continues over and over, causes self-induced rhythmic excitation of reticable tissue. Special characteristics of single transmission of nerve transmission. Myelinated and unmyelinated nerve fibers shown across a section of a typical small nerve, revealing many large nerve fibers constitute most of the cross-section area. However, a more careful look reveals many more small fibers lying in between the large ones. The large fibers may myelinated and the small ones unmyelinated. The average nerve trunk consists about twice as many as unmyelinated fibers as myelinated fibers. The average nerve trunk contains about twice as much as unmyelinated fibers as myelinated fibers. Illustrates schematically the features of a typical myelinated fiber. The central core of fiber is the axon, and the membrane of the axon is membrane that actually conducts the action potential. The action is filled in the center with exoplasm, which is viscous intracellular fluid. Surrounding the axon is a myelin sheath. That is often much thicker than the axon itself, about once every 1 to 3 millimeters along its length. Length of its myelin sheath is a node of Ranvier. The myelin sheath is deposited around the axon by a Schwann cell in the following manner. The membrane of a Schwann cell first envelops in the axon. The Schwann cell then rotates around the axon many times, laying down multiple layers of a Schwann cell membrane containing the lipid substance sphingomyelin. This substance is an excellent electrical insulator that decreases ion flow the membrane about 5,000 fold. At the junction between each two successive Schwann cells along the axon, a small unit salted area only a small unisalted area only 2 to 3 micrometers in length remains while the ions still can flow with ease through the axon membrane between the intracellular fluid and between the extracellular and intracellular fluid of the axon. This area is called node of Ranvier. Saltatory conduction of myelinated fibers from the node to node. Even though almost no ions can flow through the thick myelinated sheaths of myelinated nerves, they can flow with ease through the nodes of Ranvier. Therefore, action potential occurs only at the nodes. Yet action potential are conducted from nodes to nodes by saltatory conduction. Yet the action potential are conducted from node to node by saltatory conduction, as shown in figure 5.17. That is, electrical current flow through the surrounding extracellular fluid outside the myelinated sheath, as well as through the exoplasm inside the axon from the node to node, creating excessive nodes one after another. Thus, the nerve impulses jumps from the fiber, which is the origin of the term saltatory. First, by the causing the depolarization process to jump along the interverse along the axis of the nerve fiber, This mechanism increases the velocity of nerve transmission and myelinated fiber has each about 5 to 50 fold. First, by causing the depolarization process to jump along intervals along the axis of the nerve fibers. Second, saltatory conduction conveys energy from axon because only the nodes depolarize, allowing perhaps 100 times less loss of ions that would either be necessary. The excellent insulation afforded by the myelin membrane, about 50-fold, decreases the membrane capacity and allows repolarization to occur within little transfer of ions. 100 times or less loss of ions. Velocity of conduction in nerve fibers. The velocity of action potential conduction of the nerve fibers as little as 0.25, a small average as much as 100 meters per second, more than the length of football, built in 1 second in large myelin fibers. Excitation, the process of eliciting the action potential. Basically, any factor that causes sodium ions to begin to diffuse inward through the membrane is called is sufficient numbers can set off automatic regenerative opening of the sodium channels. Excitation, the process of clinical action potential. Eliciting action potential. Basically, any factor that causes sodium ions to begin to diffuse inward through the membrane is in sufficient numbers. Peers can get off automatic regenerative opening of the sodium channels. The automatic generative opening can result from mechanical disturbance of the membrane. Chemical effects of the membrane or passage of electrochemical membrane. All these approaches are used in different proteins in the body to elicit nerve or muscle action potential. Mechanical pressure to excite sensory movement and links of the skin. Chemical and neurotransmitters to transmit glands. Signals from one neuron to another neuron brain. Electrical current to transmit signals between successive muscle cells to heart and the intestine. Excitation of nerve fibers in a negatively charged metallic core. Usually means of exciting of a nerve or a muscle in the experimental laboratory is to apply electricity to the nerve and the muscle surface through the small electrodes, one of which is negatively charged and the other positively charged. When electricity is applied in the manner the negatively manner because stimulus of the negative electrode. Remember that the action potential is stimulated by the opening of the voltage gated channels. Threshold of excitation of acute local potentials. A weak stimulus at this point causes the membrane potential to change from 70 to 65 millivolts. 1 millisecond of both stimulus. Refractory period after an action potential during which a new stimulus can be initiated. Resting membrane potential. The period during the second action potential can visit even the strong stimulus is called the absolute refractory period. The period during which a second action potential cannot be elicited even with a strong stimulus is called the absolute refractory period. The period for large myelinated nerve fibers is about 1 of 2500 second. 2500 second. Therefore, one can readily calculate the fibers can transmit maximum of about 175 impulses per second. 100 to 2500 pulses per second. Inhibition of excitability and stabilize and local anesthetics. In contrast, the factors that increase the nerve's excitability, membrane stabilizing factors can decrease excitability. For example, a rich extracellular fluid calcium ion concentration A rich extracellular fluid ion concentration decreases membrane potential to sodium ions and stimultaneous reduces excitability. Therefore, calcium ions are said to be what is called a stabilizer. Local anesthetics. Among the most important stabilizers are the main substances that clinically are as local anesthetics, including procaine and tetracaine. Most of the channels, making it much more difficult. Okay, most of these agents act directly on the activation gates of the sodium channels, making it much more difficult for these gates to open and thereby reducing membrane excitability. When excitability has been reduced to low, that the radio of action potential strength to excitability threshold, this is reduced to 10. Nerve impulses fail to pass along the anesthetized nerves. Chapter 17, Thoracic wall, intercostal muscles and the mechanisms of respiration. The thoracic wall is formed by the thoracic cage and muscles that extend between the ribs and the intercostal muscles, intrinsic muscles of the thoracic wall. However, thoracic wall is liberally covered by all over the intrinsic muscles. The thoracic cage is described in detail in chapter 15, page 193. The thoracic wall is covered from superficial to deep by skin, superficial fossa, deep fossa, and muscles. The skin covering the thoracic wall is thin on the anterior aspect, and thick on the back aspect. The distribution of hair is variable and depends on the age, sex, and race. Cutaneous nerves. The cutaneous nerve innervation in front of the thoracic is provided by the cutaneous branch of the anterior primary rami of the thoracic spinal nerve from T2 to T6. Each sequence from above downward. Thoracic nerves in sequence from thoracic 2 to thoracic 7. Thoracic nerves T2 to T6 in sequence from above downward by the thoracic 2 at the level of the second thoracic rib to the thoracic 6 G4 process. The skin above the level of the second rib is supplied by the anterior primary ramus. In sequence above the T2 and T2 at the level of the second rib to the T6 at the level of the G5 process. The skin above the level of the second rib is supplied by the anterior primary ramus of C4 via supraclavicular nerves. Primary ramus of C4 via supraclavicular nerves. Note well, the anterior rami of C6 to T1 innervates the skin of the upper limb. C5 to T1. The cutaneous innervations of the back of the thorax on either side of the midline for about 5 cm is provided by the posterior rami of the thoracic spinal nerve. Cutaneous of the back 5 cm middle line. Superficial fascia. The superficial fascia is a denser on the posterior aspect of the chest to sustain the pressure of the body when lying in the spine position. The superficial fascia on the front of the chest contains breast mammillary glands, which is rudimentary in males and well developed in adult females. The breast is described in detail in chapter 3. Deep fascia. The deep fascia is thin and ill-defined except in the pectoral region to allow the free movement of the thoracic wall during breathing. Muscles. The thoracic wall is liberally covered by the following extrinsic muscles: the muscles of the upper limb, the muscles of abdomen, and the muscles of the back. The muscles of the upper limbs are pectoralis major, pectoralis, and pectoralis minor, muscles covering the front of the thoracic wall. Serratus anterior. Nerves on the side of the thoracic wall. Muscles of abdomen. Rectus abdominis and external oblique cover the lower part of the front of the thoracic wall. Rectus abdominis and external oblique cover the lower part of the front of the thoracic wall. Muscles of the back. Trapezius and latissimus dorsi. Trapezius and latissimus dorsi. Levator, scalpi, rhomboideus, major and minor. Trapezius, latissimus dorsi, levator, scalpi, rhomboideus, major and minor. Serratus posterior superior and serratus posterior inferior. Erector spinae. The thoracic wall is more or less completely covered by the extrinsic muscle except for the anterior and posterior medial lines. On the back, the thoracic wall is thinly covered by musculature in the region of triangle of osculation. Thoracic intrinsic muscle of the thoracic wall. The intrinsic muscle of the thoracic wall are called intercostal muscles. They are arranged in 3 layers from superficial to deep. They are as follows: external intercostal muscle, internal intercostal muscle, transverse thoracic muscle. The muscle layer is linked by the endothoracic fascia. The muscle layer is lined by the endothoracic fascia, which runs in the line by parietal pleura. These 3 layers of muscles are comparable to the 3 layers of muscles in the anterior abdomen wall. The intercostal nerves and vessels from the neurovascular bundle which lie between the intermediate and deep layers neurovascular plane. Note well, the addition of the above mentioned 3 intrinsic muscles, there is another set of muscles called levator costarum, external intercostal muscle. Each muscle extends from the intercostal space from the tubercle of the rib behind the intercostal junction. Intercostal nerves and vessels. Intercostalis intimus and collateral branches of intercostal nerves and vessels. External intercostal. Contents of the intercostal space as seen in dissection. External intercostal, internal intercostal. Intercostalis intimus. In front, it is replaced by anterior intercostal membrane. Origin insertion. It arises from the lower border of the rib above and inserts into the outer lip of the outer border of the rib below. Direction of fibers. The fibers of the external intercostal muscles are directed downward, forward, and medially. In the posterior part, however, they are directed downward and laterally. Nerve supply. By intercostal nerve of the same space. Actions. The intercostal space muscles elevate the ribs during inspiration. Internal intercostal muscle. Extend. Each muscle extends in the intercostal space from the lateral border of the sternum in front of an angle of the rib behind, which is replaced by the posterior intercostal membrane. Lateral border of the sternum in front of the angle of the rib behind sternum, where it is replaced by the posterior intercostal membrane. Origin insertion. It arises from the floor of the costal groove of the upper rib and inserts into the inner lip of the upper border of the rib below. Direction of fibers. Its fibers are directed downward, backward and laterally, i.e. the right angle of those of the external intercostal muscle. Nerve supply. By intercostal nerves of the same space. Actions. The internal intercostal muscles elevate the ribs during inspiration. Transverse thoracic muscle. The transverse thoracic muscles are divided into following 3 parts: intercostalis intimus, subcostalis and sternocostalis. Intercostalis intimus, subcostalis and sternocostalis. Intercostalis intimus extend. It occupies the middle two-fourths of the intercostal space. It occupies the middle two-fourths of the intercostal space. Origin and insertion. It arises from the inner surface of the rib upper, on the upper border of the costal group, and inserts into the inner surface of the rib below. Direction of the fibers in the same as those of internal intercostal muscles. Direction of the fibers. Nerve supply by intercostal nerve of the same space. Action. The intercostal intimi muscles elevate. Intercostal intimi lie on the same plane as the intercostal intimus muscle on the posterior part of the intercostal space. It intervenes between the intercostal nerves and the vessels and pleura. Origin and insertion. The origin of the inner surface of the rib near the angle inserts on the inner surface of the second rib and rib below. It arises from the inner surface of the rib near the angle and inserts on the inner surface of the second or the third rib below. Direction of the fibers. It is the same as that of internal intercostal muscle. By intercostal nerves. Actions. Depressor of the ribs. Sternocostalis. The sternocostalis muscle, one on either side, is situated on the inner aspect of the border on the chest wall, behind the sternum of the costal cartilage, occupying the anterior part of the upper intercostal space, except the first space. The intercostalis muscles innervanes between the anterior end of the intercostal nerves and pleura. Posterior surface of the costal lower 3 or 4 ribs. Posterior surface of the costal cartilage of the lower 3 or 4 ribs. Origin. Posterior surface of the costal cartilage of the lower 3 or 4 ribs. Insertion. The fiber diverges upward and laterally as slips to be inserted into the lower border of the inner surface of the costal cartilage of 2nd to 6th rib. The fibers diverge upward and laterally as slips to be inserted into the lower border and inner surface of the costal cartilage of 2nd to 6th ribs. Nerve supply by intercostal nerves. Action. It draws down the intercostal cartilage in which is inserted. The origin, insertion, extent, direction of the ribs of the supply, and action of the intrinsic muscles of the thorax are given in 17.1. Intercostal spaces. The space between the two adjacent ribs and their costal cartilages are known as intercostal space. The space between the two adjacent. Thus, there are 11 intercostal spaces on either side. Each typical intercostal space is directed downward and forward. It is narrow towards the vertebral column and broad towards the sternum, its widest part being a costochondral junction. The third to sixth spaces are called typical intercostal space between the blood and the nerve supply of the third to sixth intercostal space is confined only to the thorax. As they are confined only to the thorax. Typical intercostal space boundaries. The sharp lower margin of the upper rib and its cartilage. Inferior, blunt upper margin of the lower rib and its cartilage. Anterior, lateral border of the sternum, costal cartilage. Posterior, body of the corresponding thoracic vertebra. Contents of the typical intercostal space. Each space contains the following structure: 3 intercostal muscles, external intercostal, internal intercostal, innermost intercostal, intercostal nerve, intercostal arteries, intercostal veins, intercostal nymphs. Plane of neurovascular bundle in the intercostal space. The neuromuscular bundle consisting of the intercostal nerve and vessels lie between the intercostal and innermost intercostal muscles, that is, in between the intermediate and deep layers of the intercostal muscles. They are arranged in the following order from downward: intercostal vein, intercostal artery, and intercostal nerve. Mnemonic: VIN. Intercostal muscles. The intercostal muscles are a group of muscles that are present in the intercostal space and help form the chest wall. The intercostal muscles intercostal muscles constitute intercostal muscle, internal external intercostal muscle, internal intercostal muscle, innermost intercostal muscle. Nerve supply by intercostal nerves. The intercostal muscles are described in detail on page 219 to 221. The actions of intercostal muscles are as follows. They act as strong supports for the rib, preventing their separation. They act during respiration. External intercostal muscles elevate the ribs during inspiration. The actions of intercostal muscles are as follows. They act as strong supports of rib, preventing their separation. They act during respiration. External intercostal muscles elevate the ribs during inspiration. Intercostal intimus also elevate the ribs during forced inspiration. They pull down the ribs cage and push air out of the lungs. Intercostal nerves. The 12 pairs of thoracic spinal nerves supply the thoracic wall. As soon as they leave the intervertebral foramina, they divide into anterior and posterior rami. The anterior rami of the upper 11 thoracic spinal nerves, thoracic and thoracic 11, are called the intercostal nerves. As they course through the intercostal space, the anterior primary ramus of the 12 thoracic spinal nerves run in the abdominal wall below the 12th rib. Hence, it is called the subcostal nerve. Unique feature: the intercostal nerves are the anterior primary rami of the thoracic spinal nerves. They are segmental in character, unlike the anterior primary rami of other regions. Of the spinal cord and form the nerve plexus, namely cervical, branchial, lumbar, and sacral. Classification: the intercostal nerves are classified into the following two groups: typical intercostal nerves 3 to 6, and atypical intercostal nerves 1st, 2nd, 7 to 11. The typical intercostal nerves are those that remain confined their own intercostal space. Typical intercostal nerve course and relations. The typical intercostal nerve is the anterior primary ramus of the typical thoracic spinal nerve. After its origin, it turns laterally behind the sympathetic trunk and then enters into the intercostal space between the parietal pleura and posterior intercostal membrane. Parietal pleura and posterior intercostal membrane. In the costal groove, it comes in relation with the corresponding intercostal vessels and from the neurovascular bundle of intercostal space. In intercostal space, vein, artery, and nerve lie in order downward. Near the intercostal nerve of the intercostal space, the interthoracic nerve crosses the front of the interthoracic artery. Then pierces the intercostal membrane. Anterior intercostal membrane pierces the internal intercostal membrane, internal intercostal muscle, anterior intercostal membrane, and pierces and pectoralis major muscle to terminate as the anterior cutaneous nerve. Note 12. In the posterior part of the intercostal space, the intercostal nerve lies between the pleura and posterior intercostal membrane. In the remaining greater part of the intercostal space, it lies between the internal intercostal and intercostalis intimus, internal intercostal intimus muscles. Branches. Rami communicans. Each nerve communicans with the corresponding thoracic. Each nerve communicates with the corresponding thoracic sympathetic ganglion by white and gray communicants. Muscular branches. These are small slender branches from the nerve which supply intercostal muscles and serratus posterior, serratus inferior. Collateral branches arises in the posterior part of the intercostal space near the angle of the rib and runs in the lower part of the intercostal space. The upper border of the rib along the same neurovascular plane. It supplies intercostal muscles, parietal pleura, and periosteum of the rib. Lateral cutaneous branch. It arises in the posterior part of the intercostal space near the angle of the rib and and accompanies the main nerve for some distance. Then pierces the muscles of the lateral thoracic wall along the midexcellary line. It divides into anterior and posterior branches to supply the skin on the lateral thoracic wall. Anterior cutaneous branch. It is the terminal branch of the nerve which emerges on the side of the sternum. It divides into medial and lateral branches. It divides into medial and lateral branches and supplies the skin of the frontal thoracic wall. Atypical intercostal nerves. The atypical intercostal nerves are as follows. The first intercostal nerve, the greater part of this nerve, joins the ventral ramus of the cervical 8 spine from the lower trunk to form the lower trunk of the branchial plexus. The remaining part of the nerve is very small and lacks in both lateral and anterior cutaneous branches. Second intercostal nerve, its lateral cutaneous branch is called intercostobranchial nerve. Its lateral cutaneous branch is called intercostobranchial nerve. It courses across the axilla and joins the medial cutaneous branch of the arm. Cross the axilla and joins the medial cutaneous branch of the arm. The intercostal branchial nerve supplies the skin of the floor of the axilla and upper part of the medial side of the arm. In coronary artery disease, the cardiac pain is referred along this nerve to the medial side of the arm. 7th 11th thoracic nerves. These nerves leave the corresponding intercostal space to enter into the intercostal abdominal wall. Hence, they are called thoracoabdominal nerves. These nerves supply intercostal muscles of the corresponding intercostal space. In addition, they supply muscles of the anterior abdominal wall. For example, external oblique, internal oblique, and transverse abdominis and rectus abdominis muscles, and skin and parietal peritoneum covering the outer and inner surface of the abdominal wall respectively. Chapter 17, Thoracic wall, intercostal muscles and the mechanisms of respiration. Specific learning objectives. After studying this chapter, the student should be able to describe the extent, attachment, direction of fibers, nerves, supplies, and actions of intercostal muscles. Describe the type of mechanism of respiration. Give the anatomical basis of site of eruption of cold abscess on the thoracic wall, internal mammillary artery graft, and intercostal nerve block. The thoracic wall proper is formed by the thoracic cage and muscles that extend between the ribs called the intercostal muscles. Intrinsic intercostal between the ribs called intercostal muscles. Intrinsic muscle of thoracic wall. However, thoracic wall is liberally covered by all over the extrinsic muscles. This thoracic cage is described in detail in chapter 15, page 193. Coverings of the thoracic. The thoracic wall is covered from surface to deep by skin, superficial fascia, deep fascia, and muscles. Skin, superficial fascia, and deep fascia, muscles. Skin. The skin covering the thoracic wall is thin on its anterior aspect and thick on the back aspect. The distribution of hair is variable and depends on age, sex, and race. Cutaneous nerves. The cutaneous innervations in front of the thoracic is provided by cutaneous branch of the anterior primary rami of the thoracic spinal nerve from 2 to 6 in sequence from above down by the T2 at the level of the second rib and T6 at the level of the xiphoid process. The skin above the level of the second rib is supplied. The skin above the level of the second rib is supplied by the anterior primary ramus of C4 via subsupraclavicular nerves. Note well, the anterior rami of C5 to T1 innervates the skin of the upper limb. The anterior rami of C5 to T1 innervates the skin of the upper limb. So the anterior rami of C5 to T1 innervates the skin of the upper limb. The cutaneous innervation of the back of the thorax on either side of the middle for about 5 cm is provided by the posterior rami of the thoracic spinal nerve. Superficial fossa. The superficial fossa is denser on the posterior aspect of the chest to sustain the pressure of the body when lying on the spine position. The superficial fossa on the front of the chest contains breast mammary gland, which is rudimentary in males and well developed in adult females. The breast is described in detail in chapter 3. Deep fascia. The deep fascia is a thin and ill-defined except in the pectoral region to allow the free movement of the thoracic wall during breathing. Deep fascia is a thin and ill-defined except for the pectoral region to allow the free movement of the thoracic wall during breathing. Muscles. The thoracic wall is liberally covered by the following intrinsic muscles. The thoracic wall is liberally covered by the following intrinsic muscles. The muscle of the upper limb. Pectoralis major and pectoralis minor muscles cover the thoracic wall. Serratus anterior covers the side of the thoracic wall. Serratus anterior. Muscles of the abdomen. Rectus abdominis and external oblique cover the lower part in front of the thoracic wall. Muscles of the back. Trapezius and latissimus dorsi. Levator, scalpi and rhomboideus major and minor. Serratus posterior superior and serratus posterior inferior. Erector spine. Muscles of the upper limb. Pectoralis major and pectoralis minor muscles cover the front of the thoracic wall. Serratus anterior covers the side of the thoracic wall. Serratus anterior. Muscles of the abdomen. Rectus abdominis and external oblique cover the lower part. External oblique. Lower part of the thoracic wall. Muscles of the back. Trapezius, latissimus dorsi. Trapezius and latissimus dorsi. Trapezius and latissimus dorsi. Then levator, scalpi and rhomboideus major and minor. Serratus superior and serratus posterior inferior. And erector spinae. Note well, the thoracic wall is more or less completely covered by extrinsic muscle except the anterior and posterior medial lines. In the back, the thoracic wall is thinly covered by musculature in the region of the triangle of osculation. On the back, the thoracic wall is thinly covered by musculature of the region of the triangle of osculation. Interior intrinsic muscle of the thoracic wall. The intrinsic muscle of the thoracic wall are called intercostal muscles. They are arranged in 3 layers from the superficial to deep. These are as follows. The intrinsic muscles of the thoracic wall are called intercostal muscles. They are arranged from layers superficial to deep. These are as follows. External intercostal muscle, internal intercostal muscle, and transverse thoracic muscle. The muscle layer is lined by the androthoracic fascia, which is turned in lined by the parietal pleura. which is turned inlined by parietal pleura and thoracic fascia. These three layers of muscles are comparable to the three layers of muscles in the anterior abdominal wall. The intercostal nerves and vessels of the neurovascular bundle. The intercostal nerves and vessels of the neurovascular bundle lies in between intermediate and deep layers. Neurovascular plane. Note well. In addition to the above mentioned three intrinsic muscles, there is another set of muscles called levator costarum. External intercostal muscle extent. Each muscle extends in the intercostal space from the intercostal space from the tubercle of the rib behind the costochondral junction in front, where is replaced by anterior intercostal membrane. Direction of the ribs. The ribs of the external intercostal muscle. The fibers of the external intercostal muscle are directed downwards, forward and medially. In posterior part, however, the fibers are directed downward and laterally. Nerve supply by intercostal nerve of the same space. Intercostal nerve of the same space. The external intercostal muscles elevate the ribs during inspiration. The external intercostal muscles elevate the ribs during inspiration. Internal intercostal muscle. Each muscle extends in the intercostal space from the lateral border of the sternum in front of the angle of the rib, where it is replaced by the posterior intercostal muscle. Origin and insertion. It arises from the floor of the costal groove. Of the upper lip and inserts into the inner lip of the upper border of the rib below. Direction and fibers. The fibers are directed downward, backward and laterally. I.e. the right angle of the external intercostal muscle. By intercostal nerve of the same space. The intercostal muscles have relative ribs during the intercostal muscles have elevate the ribs during inspiration. The external intercostal muscles elevate the ribs during inspiration. The internal intercostal muscles elevate the ribs during inspiration. Transverse thoracic muscle. The transverse thoracic muscle is divided into the following three parts. In transverse thoracic muscle, intercostalis intimus, subcostalis and sternocostalis. The transverse thoracic muscle is divided into the following three parts: intercostalis intimus, subcostalis and sternocostalis. Intercostalis intimus, subcostalis and sternocostalis. Intercostalis intimus extent. It occupies the middle 2/4 in the intercostal space. It occupies the middle 2/4 of the intercostal space. Origin and insertion. It arises from the intercostal surface of the rib. Arises from the inner surface of the rib above upper border of the costal groove and inserts in the inner surface of the rib below. Direction of the fibers. The fibers is the same as the intercostalis intimus muscle. Subcostalis muscles. Total number of vertebrae. Intercostal muscles depress the ribs during forced expiration. Intercostal muscles depress the ribs during forced expiration. Subcostalis muscles. Total number variable. Subcostalis muscle. The subcostalis muscle lie in the same plane as the intercostalis muscle. The subcostalis muscles lie in the same plane as the intercostalis muscles. In the posterior part of the intercostal space. It intervenes between the intercostal nerve and vessels and pleura. Origin and insertion. It arises from the inner surface of the rib near the angle and inserts on the inner surface of the second or third rib below. Direction of fibers. The same as the internal intercostal muscle by intercostal nerves. Nerve supply. Actions. Depressor of the ribs. Sternocostalis. The sternocostalis muscles on either side is situated on the inner aspect of the costal wall behind the sternum and costal cartilage, occupying the internal part of the upper intercostal space, except for the first space. The intercostalis muscles intervenes between the anterior end of the intercostal nerve and pleura. Origin. It arises from the lower one-third of the posterior surface of the body of the sternum, posterior surface of the xiphoid process of the sternum, and the posterior surface of the costal cartilage of the lower 3 or 4 ribs. Insertion. The fibers diverge upwards and laterally as slips to be inserted into the lower border of the inner surface of the costal cartilage of 2nd to 6th ribs. Nerve supply. By intercostal nerves action, it draws down the costal cartilage which is inserted. The origin, insertion, extension and direction of fibers, nerve supply and actions in the intercostalis muscle of the thoracic are given in table 17.1. Intercostal space. The space between the two adjacent ribs and either costal cartilage are known as intercostal space. Thus, there are 11 intercostal spaces on either side. Each typical intercostal space is directed downward and forward. It is narrow towards the vertebral column and broad towards sternum. Its widest part being at costochondral joint. The third to sixth costal spaces are called typical intercostal space. The third to sixth spaces are called typical intercostal space because the blood and the nerve supply of the third to sixth intercostal space is confined only to the thorax. Typical intercostal space boundaries. The sharp lower margin of the upper rib and its cartilage. Superior boundaries. Intrinsic muscle of the thoracic wall. External intercostal muscle. Internal intercostal muscle. Transverse thoracis. Intercostalis intimus, subcostalis and sternocostalis. Origin, lower border of the lip, floor of the costal group, inner surface of the group. Inner surface of the near the angle. Inner surface of the rib above. Inner surface of the rib near angle. Lower one-third of the posterior surface of the sternum. Posterior surface of the xiphoid process. Posterior surface and posterior surface. Floor of the costal group. Lower border of the costal group. Insertions. Outer lip, inner surface and costal cartilages. Extent, direction of the fibers, nerve supply and actions. Boundaries. The sharp lower margin of the upper rib and its cartilage. Boundaries. Superior. The sharp lower margin of the upper rib. Inferior. Blunt upper margin of the lower rib and its cartilage. Sharp lower border of the upper rib. Superior boundary. Inferior boundary. Blurred upper margin of the lower rib and its cartilage. Anterior lateral border of the sternum between the costal notch. Lateral border of the sternum between the costal notch. Body of the corresponding thoracic vertebra. Posterior. So superior, inferior, anterior, posterior. Contents of the atypical intercostal space. Each space contains the following structures. 3 intercostal muscles. External intercostalis, internal intercostal and innermost intercostal or intercostalis intimus. Intercostal nerve, Intercostal arteries, one posterior, two anterior. Intercostal arteries, one posterior, two anterior. Intercostal arteries, one posterior, two anterior. Intercostal veins, one posterior, two anterior. Intercostal love vessels and lymphoid nodes. Note 12. Plane of neurovascular bundle in the intercostal space. The neurovascular bundle consists of intercostal nerve and vessels lying in between the internal intercostal and innermost intercostal muscles, that is, between the intermediate and deep layers of intercostal muscles. And they are arranged in the following order from above downward: intercostal vein, intercostal artery, and intercostal nerve. Mnemonic VAN. Intercostal muscles. The intercostal muscles are a group of muscles that are present in the intercostal space and help form and move the chest wall. The following muscles constitute intercostal muscles: internal intercostal muscle, external intercostal muscle, and innermost intercostal muscle, or intercostalis intimus. Nerve supply by intercostal nerves. The intercostal muscles are described in detail in page 193 to 221. The third to the sixth space are called typical intercostal space because the blood and the nerve supply of third to sixth intercostal space is confined only to the thorax. Actions. The actions of intercostal muscles are as follows. They act as strong support for the rib, preventing their separation. They act as strong support for the rib, preventing their separation. They act during respiration. External intercostal muscles elevate the ribs during the inspiration. Internal intercostal muscles elevate the ring during forced inspiration. During forced inspiration, they pull down the rib cage and push air out of the lungs. Intercostal nerves. Intercostal nerves. The 12 pairs of thoracic spinal nerves supply the thoracic wall. As soon as they leave the intervertebral foramina, they divide into anterior and posterior rami. The anterior primary rami of the upper 11 thoracic spinal nerves are called intercostal nerves as they course through the intercostal space. The anterior primary ramus of the 12th spinal nerve runs in the abdominal wall below the 12th rib, hence it is called the subcostal nerve. Unique features. The intercostal nerves are the anterior primary rami of the thoracic spinal nerves. They are segmental in character, unlike the anterior primary rami. The intercostal nerves are the interior primary rami of the thoracic spinal nerve. They are segmental in the character, unlike the anterior primary rami, from other regions of the spinal cord that form nerve plexus, namely cervical, branchial, lumbar, and sacral. Classification. The intercostal nerves are classified into the following two groups: typical intercostal nerves, 3rd to 6th, and the atypical intercostal nerves, from 1st, 2nd, 7th to 11th. The typical intercostal nerves are those that remain confined in their own intercostal space. Typical thoracic spinal nerve. GR, gray rami communicates. WR, white rami communicates. Lateral muscular branch. Medial sensory branch. Medial coteus branch. LCB, lateral coteus branch. Sympathetic ganglion, collateral branch. Sympathetic ganglion, collateral branch. WR, white rami communicates, and GR, gray rami communicates. So here we see the spinal cord, anterior root and posterior root. The anterior root and the posterior root meet together. Before the posterior root meets, it meets with the dorsal root ganglion. After meeting, it again divides into two parts. In the anterior, it becomes the anterior primary ramus, and in the posterior it becomes the posterior primary ramus. Then the posterior primary ramus divides and subdivides in the back, and the anterior primary ramus continues to become the anterior cutaneous branch. The cutaneous branch then becomes lateral cutaneous branch and it comes closer to the outer skin. The atypical spinal nerves extend beyond the thoracic wall and partially or entirely supply other regions. Atypical intercostal nerve, course and relations. The atypical intercostal nerve is the anterior primary ramus of the typical intercostal nerve is the anterior primary ramus of the typical thoracic spinal nerve. Course and relations. Typical intercostal. The typical intercostal nerve is the anterior primary ramus of the typical thoracic spinal nerve. After its origin, it turns laterally behind the sympathetic trunk, and then enters into the intercostal space between the parietal pleura and the costal intercostal membrane. Then it enters the costal groove of the corresponding rib to course laterally and forward. It then enters the costal groove. It then enters the costal groove of the corresponding rib to course laterally and forwards. In the costal groove, it comes into relation with the corresponding intercostal vessel and forms the neurovascular bundle of the intercostal space. In the intercostal space, vein, artery, and nerve lie in order from above downward. Near the sternal end, the intercostal space, the intercostal nerve crosses in front of the internal thoracic artery. Then it pierces internal intercostal muscle, anterior intercostal membrane, and pectoralis major muscle to terminate as the anterior cutaneous nerve. In the posterior part of the intercostal space, the intercostal nerve lies in between the pleura and the posterior intercostal membrane. In the membrane greater part of the intercostal space, remaining greater part of the intercostal space, it lies between the internal intercostal and intercostal muscle. In the remaining greater part of the costal space, in the posterior intercostal nerve lies between pleura and posterior intercostal membrane. Intercostal intimus. Branches. Rami communicants. Each nerve communicates with the corresponding thoracic sympathetic ganglion by white and gray rami communicants. Rami communicants. Each nerve communicates with the corresponding thoracic sympathetic ganglion by white and gray rami communicants. Muscle branches. These are small, slender branches from the nerve which supply intercostal muscle and serratus posterior, superior and inferior. These are small, slender branches from the nerve which supply intercostal muscles and serratus posterior and serratus inferior. Collateral branch. It rises in the posterior part of the intercostal space near the angle of the rib. These are small, slender branches from the nerve which supply intercostal muscle and serratus posterior. Supply intercostal muscle branches. So branches of the nerve are rami communicates and muscle branches. From the nerve, from supply intercostal muscle, intercostal muscle and serratus posterior, superior and inferior. Collateral branches. It arises in the posterior part of the intercostal space. In the posterior part of the intercostal space near the angle of the rib and runs in the lower part of the space along the upper border of the rib below the same neurovascular plane. Along the upper border of the rib in the same neurovascular plane. Collateral branch. It supplies intercostal muscles, parietal pleura and periosteum of the rib. Lateral costal branch. It arises in the posterior part of the intercostal space. It arises in the posterior part of the intercostal space near the angle of the rib. Lateral LCB. Lateral cutaneous branch. Lateral cutaneous branch. It rises in the posterior space angle of the rib and accompanies the main nerve for some distance, then pierces the muscles of the lateral thoracic wall along the medullary line. It divides into anterior and posterior branches and supplies the skin and lateral thoracic wall. Lateral branch and collateral branch. Collateral branch. It arises in the posterior part of the intercostal space near the ankle of the rib and runs in the lower part of the space along the upper border of the rib along the same neurovascular collateral neurovascular plane. It supplies intercostal muscles, parietal pleura and periosteum of the rib. Lateral cutaneous branch. It arises in the posterior part of the intercostal space near the angle of the rib and accompanies the main nerve for some distance, then pierces the muscle of the lateral thoracic wall along the midixillary line. It divides into anterior and posterior branches and supplies the skin and lateral thoracic wall. Anterior cutaneous branch. It is the terminal branch of the nerve which emerges on the side of the sternum. It divides into medial and lateral branches. Divides into medial and lateral branches and supplies the skin of the front of the thoracic wall. Atypical intercostal nerves. The atypical intercostal nerves are as follows. First, intercostal nerve. The greater part of this nerve joins the ventral ramus. The greater part of this nerve joins the ventral ramus. Cervical 8 spinal nerve from the lower trunk of the branchial plexus. Cervical 8 spinal nerve lower frontal plexus. The remaining part of the nerve is very small and it lacks both lateral and anterior cutaneous branches. Second, intercostal nerve. Its lateral cutaneous branch is called intercostobrachial nerve. It courses across the axillary and joins the medial cutaneous branch of the arm. The intercostal branch nerve supplies the skin of the floor of the axilla and upper part of the medial side of the arm. In coronary artery disease, the cardiac pain is referred along this nerve to the medial side of the arm. 7th to 11th intercostal nerves. These nerves leave the corresponding intercostal space to enter into the intercostal abdominal wall. Hence, they are called thoracoabdominal nerves. These nerves supply intercostal muscles of the corresponding intercostal space. In addition, they supply muscle of the anterior abdominal wall, for example, internal oblique, external oblique, internal oblique. Muscles of the anterior abdominal wall, for example, external external oblique, internal oblique, transverse abdominis, and rectus abdominis muscles, and skin and parietal peritoneum covering the outer inner surface of the abdominal wall respectively. Clinical correlation. Root